I welcome you to the next tour worldwide, as Marina said yesterday, to hear our presentations, and we will start in South America. I invite Nicola Fiore to give his presentation of main phytoplasma infecting crops in South America.
Thank you, Wolfgang. Good morning to everybody. I will find my presentation. No, it's not mine. The other section. Okay, this is the right presentation. Okay. Thank you so much to the organizer of this Congress, Ali Assunta, for inviting me for this presentation. And really, the phytoplasmas in South America we have several phytoplasma infected several crops. But I think that is necessary to have more study about this topic, principally in epidemiological study. In the central zone, normally around the tropic, maybe there are some problem related to phytoplasma, but we have no information generally. This is a little review. Of course, it's not be complete because the information is a lot. But only the main phytoplasmas have been considered by me, and the most widespread, the most prevalent, and the one that can cause most damage in different crops. I anticipate my apology. Itself is not mentioned in my presentation, but I hope in the future, in the proximate future, have the opportunity to make a big review as the colleague of India made with three fantastic books that were presented yesterday. We may begin with a review general involving the other colleagues that are working in phytoplasma topic in South America. Well, I know I can use this one better. We have in South America Candidato phytoplasma asteris, of course, is one of the phytoplasma presents in this country. One of them is the 1B, and we have chlorotic streaking in mice, chlorosis, dwarfing, especially in Colombia and Peru. In Chile, we didn't found phytoplasmas associated to corn generally. Lethal white in oil palm in Colombia, and then brotes grande, big bud, potato in Bolivia, a sugarcane yellow leaf syndrome in Brazil, associated to 6CSR1B. Then the other phytoplasma also belonging to the ribosomal group 1, were found in Colombia, Brazil, and Peru in different crops: carrots with yellow, bronzing, dwarfing, corn with yellowing, proliferation, deformation, and reddish plant. Is so easy to recognize reddish plant, little reddish plant in the field that is infected for one of the phytoplasma belonging to the group 1, not better specify. Coconut with yellowing, then papaya with yellowing, stunting and proliferation, and then potato with yellowing, stunting and lack of tuber production. Then we go to Chile and Argentina, and we have this very serious problem in strawberry. It's a tremendous phyllodes associated with strawberry, and the phytoplasma involved or associated is Candidata-Phytoplasma hispanicum, specifically the group, the 13 group, subgroup, ribosomal group F. You can see the symptom, this phytoplasma is induced in strawberry, and this is a problem that we are living in Chile and Argentina. Most of these plants are eliminated from the field. I want to tell you that this Candidata-Phytoplasma hispanicum and Candidata-Phytoplasma meliae are only present in American continent at the moment. Then in Chile, also in Chile, we found the 60SR13F phytoplasma in mandarin and sweet orange. And you can see the proliferation, which is broom, really, and the formation and rolling of the leaves in mandarin and sweet orange. In both cases, the study that we made in epidemiological studies that we are going on, actually in strawberry and mandarin, sweet orange, we found a succeed associated present in the field of strawberry infected and of oranges, sweet oranges infected. A first transmission trial on periwinkle with this insect was induced the appearance of phyllodes in the viruses in periwinkle plants. Then the problem now, the problem generally in South America is the identification of the insect vector, because we have a lot of this individual that we found in the field that may classificate identify at the level of genera, but not at level of species. And 99% of them probably are new species that will be classified soon. We are working on this soon. The other phytoplasma is the 6E-SR13C, detected in China trees, in Argentina, Bolivia, Paraguay, and the other one, 13E, in papaya, which has or shows symptoms of chlorosis and apical curl necrosis of the fruits. In Brazil, in broccoli plant, we have another group, the same group, not specify the subgroup, ribosomal subgroup, with maybe is associated to stunted plant, reddening leaves, and necrosis of the phloem welsel. In this plant, in infection, mixed infection or simple infection, we have to the group 1 and the group 3 in some cases. The other important phytoplasma in South America found only in Argentina, in China trees again, is Paraíso plant, is paradise plant. The plants present yellowing, as you can see, and the difference of the leaves from infected plant and healthy plant. And this is a transmission by graft that show the symptoms in plant grafted. Of course, in some cases associated to China trees, in Argentina, they have the phytoplasma, Candidata-Phytoplasma asteris. Also, the Candidata-Phytoplasma meliae is always in Argentina, was detected in plum plants. The disease, the name of the disease, a plum jello, which is blooming, and in some cases, it's possible to find a mixed infection with the 6ESR3b phytoplasma. You can see the short internode, the rolling of the leaves, the reddish leaves, etc. Then we are jumping to another candidatus, Candidato Phytoplasma fraxini, that we found associated to Unimoline. In Spanish is named Murtilla. This is a bush that grows spontaneously in the south of the country, and is very important because produce a berry that are very, very, very good, very nice. And when the phytoplasma 7A and the other one that 5A is present in this plant, the plants show witch's broom. The fruit has little and without color and without flower, generally. And then the same phytoplasma was detected in peony plants, always in Chile, with a hard deformation of the leaves generally, but the most problematic things is the virescence in the flower, as you can see. Always in the group 7, the 7B it was found in Brazil in a region plantains and Cataranthus roseus, and then the 7C in alfalfa, Argentina strawberry. The strawberry, I show you the phyllogy symptoms associated strawberry together with the same symptom associated to the presence of the phytoplasma 13F. But in these cases, there is a photo picture of alfalfa with stunt, yellowing, and short internode. In Brazil, in the region of Bonariensis, was detected the phytoplasma 7D. Come back to Chile, Uruguay, Argentina. We, in this country, Candidato Phytoplasma piri was found. I showed you yesterday in my presentation the symptoms of Candidato Phytoplasma piri, the 6ESR10C phytoplasma, and the other very news that it was detected so in peach plant in Argentina, no? And as I see you yesterday, in Chile, there is not the presence of Cacopsylla piri or Cacopsylla pericola, the main vector of this phytoplasma. Only in Argentina was detected Cacopsylla piri, and may be a problem in the transmission of this phytoplasma. The symptoms I showed yesterday, necrosis of the vessel, epinasty, low new production of fruit. These are the symptoms associated to peach plants, yellowing, leaf deformation, rolling of the leaves, really production zero, absolutely. And it was found only in Argentina associated to peach plant. But this is the king of the phytoplasma present in South America, is Candidato Phytoplasma pruni. The group is 6ESR3. The most widespread phytoplasma between this group is the 3J subgroup. We found it historically in sugar beet initially. This was a great problem in Chile, in the south of Chile, in which the crop is very important. And these are the symptoms with yellowing, and this is a yellow-white disease, then deformation and rolling of the leaves, and near to summer, the appearance of the necrosis in the leaves. Of course, the roots is inexistent in this case. But 3J was found in Chile in carrot plants, in Swiss chard, lettuce, potato plant, and then also in cactus pear. This is very interesting, this kind of witch's broom in cactus pear. Then we found 3J on cherry plants. You can see there the reducing of the growth of the plant until the death of the same. And this is a problem in some cherry plant in some area of the country. The other crops infected by phytoplasma are grapevines. And the symptoms are no different from when associated to other kind of phytoplasma or the species of phytoplasma present in the plant. And this is the 3J in grapevine in Chile. It's a very, very important problem in grapevine. I think that this, the most widespread phytoplasma associated to the grapevine in Chile. And for this reason, we decided to make an epidemiological study. And this is the case of one of the vineyards that we visited, in which we saw the symptoms of grapevine yellow in the grapevine plants. Then we found the phytoplasma in weeds associated, present in the vineyard or in the vicinity of the vineyard. And then we found the most prevalent insect, also positive to the same phytoplasma by analysis, that is a Cicadelide, Paratanus exitiosus. Paratanus exitiosus is widespread not only in Chile but in Argentina too, or where we have information for the country for which we have some information. But I think that is one of the most important Cicadelide spread all over South America, at least in the south part of South America. Then, thanks to the Tropic Safe project leading by Assunta, we carried out the transmission trials. These transmission trials in which we found during our research another widespread Cicadelide, that is Bergallia valdiviana, the other one, Paratanus exitiosus and Bergallia valdiviana. We performed the transmission trials in periwinkle and in grapevine plants. And these are the results on periwinkle. The symptoms appears in tree at maximum 6 months from the feeding. And you can see all the symptoms associated to periwinkle plant infected by the insect. The phyllogy very clear in this case. The difference between infected plant and healthy plant of periwinkle. Then, but after 1 year, or in some cases 2 years from the feeding, we may detect the phytoplasma and see so symptoms in grapevine plant too. And the principal symptoms are deformation, leaves deformation, short internode, and then the producing of 2 axes from the same node. The plants were positive in grapevine. The transmission works so in also in grapevine plant, but we have to wait in more time to see the results. Then, always in the frame of Tropic Safe, we following to search for insect and found 5 new, found more insect, all Cicadelids, positive to the 3J. One of these, we don't know the species. Maybe it is another insect that we should classify soon. Other plant that work as a reservoir of the phytoplasma associated to the vineyard or present in the vineyard on the vicinity. And then we performed transmission trials, but this time for the great quantity of individual or species found, we use in vitro transmission trials. It is very, very good result. The insects are not stressed because the humidity is 10% constantly present in the environmental, in the tube. We put the tube and the other side upside down because the insect come until the light, and then we found the leaves and then feed. And it's true that more of these plants are contaminated because we lose this plant because the medium may contaminate it for the presence of the insect, but we have the possibility to make a lot of trials in the reduced space and with the environmental condition very, very controlled in BRF, for example. And then the balance is positive. We have a lot of plants that may acclimate with acclimatation, did this work, and then appears the symptoms later, in this case in pink. Well, we are performing epidemiological study in pear, in some fields, in some orchard infected by pear decline phytoplasma. And then we soon we present new results about this epidemiological study. That's very, very important. We, about Candida phytoplasma piri, I tell you that we found Cacopsylla bidens, not too much associated to pear plants, but other psyllid that feeding constantly in pear plants. We found it in the leaves of pear plants. And this is very important because the most probably situation that this new psyllid, we don't know, maybe other, we need other run of classification, this kind of psyllids. Maybe this has the phytoplasma that move the insect that move the phytoplasma, Candida phytoplasma piri, in the orchard in Chilean fields. Concluding my speech, the most widespread phytoplasma in South America is Candida phytoplasma pruni, especially the group 3, subgroup J, is maybe is the situation at the moment. But I think it is necessary to inform producers more frequently. It is important stay closer to the industry to show the situation. Maybe most of them, I think 99.99% do not know the phytoplasma and the damage that cake causes. And then normally these symptoms are associated to nutrition problem, water problem, other environmental problem, I don't know, fungus, but never they think about phytoplasma. This is very important to do more information to the producer, because if they do not know the problem, cannot act correctly. This is the point. It's very important to expand the monitoring. The monitoring has to be constant, always. And maybe in that country where the research on phytoplasma is not moving too fast as in Chile or Argentina or Colombia or Brazil, for example. The other important point are the epidemiological studies. I'm very happy to participate to Tropic Safe because it allows to complete the epidemiological study in grapevine in Chile. And it's very important to know which are the vector, which are the reservoir plant to control adequately the disease associated to these phytoplasmas. And then, not the least, but only the last in the list, to implement the certification program. This is lack not only in South America. This is a problem, I think, in all the world, because phytoplasma are considered as pathogens of second category. I don't know why, but the problem are very high. We in this day saw a lot of picture, a lot of situation. Yesterday, with our other colleagues, we saw the great problem associated to phytoplasma and what is mean about the industry as economic consequence. And then we have to work very hard to implement the certification program worldwide. It's very important. Thank you so much for your attention. I am here for eventually questions. Thank you.
So, thank you, Nicola, for being perfect in time. So you have time for questions. Are there questions? Barbara?
Thank you, Nicola, for the presentation. In the first slide where you showed the grapevine plants from the transmission trials, you mentioned that it took 2 years, if I really understood, that they showed symptoms.
Yes, in grapevine, only in
yes,
grapevine.
Yes, no,
in periwinkle, 6 months.
No, I'm talking about grapevine.
Yeah.
And did these plants also come from in vitro cultures?
No, this was micropropagated. Because we in the laboratory have an in vitro. We performed the sanitization of plant concerning the viruses, and we are producing healthy plant for some nursery, and we have the plant free of pathogens, most important pathogens. But we saw that some species of virus, for example, grapevine rupescens stampede associated virus, we may eliminate this virus from the grapevine plant after the sanitation program. Phytoplasma bacteria and the other fungi in the wood are eliminated. So, and then we use healthy plant obtained by micropropagation, but then acclimated and in soil. The first transmission trials were in plant in cage. The last one were in vitro.
And after this incubation, so you maintained them at least for these 2 years?
Yes.
Insect
yes.
proof conditions
yes.
and
yes.
Using insecticides or yes?
Yes. No, it's all under control.
Yes. Do you have an idea for these belated symptom expression?
Sorry, I don't.
Do you have an idea why this symptom expression is so belated?
The problem. No. The thing is that normally the symptoms. Normally, I saw the symptoms in the field, in these cases, appear 1 year later by the feedings of the vet instead of in the field. Okay, we start to see symptoms in controlled condition 1 year, 1.5 year to here, but the most 2 years. I think it is normal. It's okay. In grapevine. I don't know if you have another experience, but I would know if you may observe symptoms yearly. It will be interesting. But you have to think that this is a 3J phytoplasma. It's not bois noir, it's not flavescent doré. Maybe the difference is this. If you trials, you observe symptoms yearly.
So thank you. Maybe you can discuss the specific points in the coffee break. Are there other questions for the broad range you presented here? Sandrine?
Morning. Perhaps I missed the information, but plus près? Okay. So morning. Perhaps I missed the information, but how long did the insect stay on the little in vitro plantlet?
You mean in vitro trials? Okay. Really, in plants, normally the insect die at the most 7 days before the start in plants, before the start of the trials. In vitro, we have to put before because they finished eating all the plant. But they are living very, very well in this condition. This is the strengthness of this technique because the insects are very comfortable in this condition. We have to put out there the tube at least no more than 7 days because the symptoms, they can destroy the plant if you leave in feed.
But then do you have problems of contaminations? Don't you have problems of contaminations?
Yes, I told him we have problem with the contamination medium, but as we can do it a lot of trials, a lot of tube, we have a lot of plant acclimated after. And then the balance is very, very positive, at least.
So there's one more question.
Yeah, good morning. Do you hear me? Do you hear me? Yeah. I don't know if you already said it, but is the origin of the Phytoplasma peri subgroup G is known, and the leafhoppers you identified as being vectors are like exogenous or invasive, or are they native from South America? Yeah, the origin of the Phytoplasma, and if the leafhoppers are exogenous or invasive, or if they're native from South America.
No, these are native. The 3J is absolutely a Phytoplasma that born, I don't know, but in South America generally. And then the insect we are found, the insect vector that we are found in the field, so are originary of South America, because some of them there are no description. Absolutely, there are no description of them in the literature. We have to classify some of them. We are making the barcoding. We at least may know the genera, but the species, and some I show you something, but there is much more of this.
Okay, thank you. Time is running. It was a very nice presentation overview.
Thank you.
So we are coming back to this country, to Oman, and I invite Ali Shoubi to present his first report of 16 SR2D phytoplasmas in Verbena and Chilioides, showing phyllid in Oman.
Good morning. Assalamu alaikum. Today I will give a presentation about first report of Fabricia eclodesis, known as golden crown bread in Oman. And this is the first time we register it. And in the first time I will give general about how phytoplasma situation in Oman. So phytoplasma in Oman. First phytoplasma we discovered it or recorded in Oman is lime wilshire broom. During early 1970s, there is a survey about Lyme disease in Oman, about disease in Oman in the Bautna region, and they found the symptoms of the phytoplasma. In that time, of course, there is no person known this is phytoplasma or virus. So in that time, they registered as virus. From that time until now, we register more than 30 in the economic crops, plus there is more than 10 or 20 in wild plant and weeds also. If we come to see how many phytoplasma in Oman we have, until now we register 6 types, and mostly from group 2. We've registered from Lyme, group 2B, and then also we register from the alpha-alpha phytoplasma, group 2D, and this is more aggressive than others because we register from most of the crops in Oman. And also we register a candidate from economist spines from group 6. Candidate phytoplasma omanizes from group 29. And also we have from group 6. This is Truffoli. And the final one, we candidate phytoplasma group 2W. And we do not find any symptoms or from south, especially in Lyme. In the south area, we do not find it in Lyme as a symptoms, clear symptoms. But in the about group 2D, we found it clear symptoms in the south. Now we come to about candidate phytoplasma of orthoporia in Oman. Of course, we already speak about this. Now more than 80% or 85% of Lyme in Oman is destroyed because of this disease. In 1995, the Buffet group will register it as give it a name as a candidate phytoplasma orthopolia. Then has been recorded in UAE in 1980s, from Iran 1990s, India 1999, first recorded, Saudi Arabia 2009, and then from Brazil 2015. Phytoplasma group 2D in Oman, this is of course will spread a lot. So we found it first in alpha-alpha. We studied in alpha-alpha. The symptoms will be phyllody in the early stage, and the final stage will be standing and bushy. And the sesame will find it as a fluorescent and phyllodes. And also in chickpeas will be fluorescent or phyllodes. Carrot, we find it as phyllodes. And also in eggplant, we find it phyllodes. In Spanish, we find it phyllodes symptoms. Here, field bees, we find it as phyllodes. Faba bean, phyllodes. And this is rocket, we'll find it fluorescent symptoms. Here in squash, we find it in male flower will be fluorescent, and phyllodes will be in female flower. This tomato will be phyllodes. During last year, we found during our one of the survey, we found the Fabriciata enclosure is infected with phytoplasma phyllodes. And actually, this plant usually in traditional medicine, especially in India. And also, some study from India said this is used as a diabetes. The flower and plant, for instance, are visiting by honeybee for nectar collector and also for feed from other insects like moth, butterfly. This is during we find it in the Saline Natural Park, and this is look like in totally isolated area from, there is nothing surrounding by farm because this is a natural park, and they use it as a ornamental. And this is the symptoms. We see already phyllodes, and this is the healthy one, and this is sometimes we see some bushy or witch's broom. So we studied molecular by using 4 marker, 16S RNA, sRNAs, CKA, MB, and SAB11. The result from the phylogenetic tree from 16S, we find it almost will be cluster with the 16S, 16 group 2D. CKA gene also, we cluster with the gene group 2D. IMB gene also clustered with the group 2D. And when we come to SAB11, we found it there is one mutation between group SAB11 of this plant and also group 2D, one mutation, but it's also closer to group A, identical. And usually group D and A almost so close together actually. And when I go to see if it is a truth mutation or only silent. I found it silent because amino acid sequence, it is showing identical between group 2D and this phytoplasma. So that means this is silent mutation. So main finding here will be, for instance, showing Fallot disease symptom typical to phytoplasma infected are similar to what called sesame phyllodes in Oman. DNA sequence of all the 4 phytoplasma of this phytoplasma is isolated, show identical with all of the 4 genes, except in SAB11 we have one as isolate is all same, identical. Phylogenetic tree of 4 genes using phytoplasma is linear with the group 2D, as we see. And this is first time registered from this plant infected with the group 2D. In India, they register it as a phytoplasma infected, but they do not study it, which group, only just prove it this is phytoplasma, but without it. But in Israel, 2004, they registered it from group 6, Trovoli. It's not from group 2D. 16SD subgroup phytoplasma are more aggressive than other because in Oman we spread a lot, so we can, even not in Oman, in UAE I think, and also in Saudi Arabia is registered group 2D, and in India, Iran, group D registered from a lot of hosts. At the end, I want to give my thanks to my colleagues, Prof. Abdullah Saadi, Prof. Rashid Saadi, and Ms. Alia Al-Alawi, and also for Sultan Qaboos University for their funding. شكرا جزيلا.
So, thank you, Ali. It was a short presentation, so we have time for questions. I will do it.
Thank you, Dr. Sobi. Clearly, your presentation demonstrated that you have again a wide prevalence of second D phytoplasma in Oman. Did you identify any potential vector? Because potential vector species, either leaf hopper or plant hopper, responsible for transmission.
The mutative factor, what will in alpha usually all time we find Embosca species. And also when I make a Eulotroph in sesame, also Embosca. The Embosca is the main one. We expected there will be mutative factor for this group 2D. But for lime will be Hashimonas.
Because there must be some potential vector for wide node distribution and natural distribution of this particular phytoplasma strain in Oman.
Yeah, the Embosca, I found it in south also, in the field of alfalfa infected. For that reason, the symptoms will be in the desert, it is showing because of there is a factor. But in lime, until now, I do not register it as a symptom. I thought because there is no Hashimonas there. I don't find it until now.
Thank you.
So, are there further questions? I can move around. No?
Thank you, Ali, for informative presentation. You didn't mention about the lethal yellowing in date palms or the infection in sugarcane as well?
Regarding the fellow date palm, already we registered, but I don't put it in slide here because we have, as I mentioned for you, we have more than 30 plants is infected with group 2D. Only I guess represented one. About sugarcane, already I deducted on it as a but I know until now I don't publish it. Date palm. About sugarcane, sorry.
Thank you.
And also we detected in garlic and others ornamental, but that time do not have to publish it.
There's a last question.
Dr. Thank you for the presentation. Just I want to ask about the percentage of the infection on the area during the survey. Percentage.
Actually, I do not study the percentage there because only one time I visit there for other purpose actually, but because phytoplasm, so that means I catch it. But it is look like more than 50%. And that area, you know, Salil Natural Park is isolated totally from the farm near to by that. This one, group 2D. Sugarcane, I expect 2D also, according to primary result what I have it.
Are there questions online, Fabio? No, nothing. No further questions? Okay, then thank you, Ali. And we go. I invite Gulnara Balkisheva for an historic perspective of the phytoplasma diseases in Azerbaijan.
Thank you, Wolfgang, and good morning, everybody. Now I would like to present phytoplasma disease in Azerbaijan and historical perspective. The studies on phytoplasma disease started with the collaboration of French colleagues from INRA Bordeaux Center, and the molecular characterization of the phytoplasma disease started in 2003 when French colleagues from INRA Bordeaux were in Azerbaijan to conduct the surveys. And the first surveys conducted to Guba and Absheron regions of Azerbaijan, and then we have the large crop surveys in different regions, as well as Khachmaz, Shaki, Ismailli, Genge, Gebele, Gazakh, and also Nakhchivan. I would like to say that the first surveys showed that in Azerbaijan, the Candidatus phytoplasma solani, Candidatus phytoplasma piri, prinorum, and Brazilians were present. And we have found these phytoplasmas in grapevine, cherry, peach, pear, apricot, cherry plum, and medlar tree. Also in the eggplant, pepper, and tomato from Absheron Peninsula, Guba, Shaki, Genge, and Guba regions of Azerbaijan. This is all of the phytoplasmas found in Azerbaijan. And now I would like to say how we performed the research on phytoplasmas. So first, we have performed first report of the Candidatus phytoplasma brasilians in Eurasia, and this phytoplasma was a new phytoplasma. As you know, this phytoplasma found in 2001 in Brazil, in hibiscus. But then we found this phytoplasma in Guba, in peach tree. This is the first report of this phytoplasma, other than hibiscus and other way than Brazil. And as it was interesting for us, we have we isolate the Candidatus phytoplasma brasilians genes by comparative RAPD and cloned and the sequenced the DNK locus. And we have designed the PCR, nested PCR, based on DNK gene. And you can see the primers designed. And this nested PCR allow us to differentiate two genetic distinct two genotypes, different, which is which one hibiscus witch's broom in Brazil, and the other genotype was Suriname virescens in Periwinkle, and basil from Lebanon, and peach from Azerbaijan. And then we have found the Phytoplasma candidatus prunerum on apricot, on plum tree, and Candidatus Phytoplasma piri on declined pear tree. And we was performed the multilocus sequence analysis on the Sekigrig, on 4 gene, Sekigrig, ASF, PNP, and AMP, and where the Candidatus Phytoplasma prunerum was well classified in the Candidatus Phytoplasma prunerum cluster. The Candidatus Phytoplasma piri isolate from peach tree showed interesting results as these Phytoplasma isolates gave the shared the some alleles with the Candidatus Phytoplasma piri. You can see here for the this is Phytoplasma Candidatus piri from the reddening pear tree, and this Candidatus Phytoplasma piri isolate Sekigrig, ASF, and PMP genes was as Candidatus Phytoplasma piri clustered in the as Candidatus Phytoplasma piri, and for the EMP gene they have I'm so sorry, for the PNP gene they have allele with the and clustered with the Candidatus Phytoplasma prunerum. So we can suggest that this is the interspecies recombination between Candidatus Phytoplasma piri and Candidatus Phytoplasma prunerum. For the group 12, for the Candidatus Phytoplasma solani, we have found these phytoplasmas on the common medlar tree, cherry tree, as well as eggplant, pepper, and tomatoes. And the first report of these phytoplasma associated with bois noir in Azerbaijan and grapevine have been reported in 2016. It was reported from the Gabala and Absheron regions of Azerbaijan, and the symptoms were the leaf reddening, leaf rolling, and irregular lignification. And we have, and also insect collection was done in Guba and Absheron region. The insects insects infected was Hyelestes obsoletus, identified as Hyelestes obsoletus, and Reptalus noahi. We have all of the Candidatus Phytoplasma solani isolates from Azerbaijan was subjected to the multilocus sequence analysis with two Sekigrig, MLP1, which developed by us, and then and also stamp. stamp gene. And we can see that stamp gene was most various, and in Azerbaijan, and the insect vectors have two different two clusters, grouped in two clusters, different each one others. Also, the grapevine isolates. With grapevine isolates, we have the finer differentiation with MLP1 gene. As with this gene, we have the 5 different genotypes between the Azerbaijani isolates. In the conclusion, I would like to say that in Azerbaijan we have found the Candidatus phytoplasma piri, Candidatus phytoplasma prunurum, Candidatus phytoplasma solani, Candidatus phytoplasma brasiliense. And the most widespread phytoplasma in Azerbaijan was the Candidatus phytoplasma solani. And this work financed by different projects, and we would like to express special gratitude to and thank to Dr. Ahmed Keypour, and also to colleagues from France Bordeaux Center, INRA Bordeaux Center. And this review is dedicated to the memory of Professor Jalal Aliyev and Jean-Luc Denet, who passed away. Thank you for your attention.
So thank you, Gulnara. There's time for questions. There's one, Kadriye.
Thank you, Gulnara, for your presentation. I was wondering what was the incidence of Candidatus phytoplasma brasiliensis in the peach trees? Did you make any survey?
Yes. The Candidatus phytoplasma brasiliense we have found during our surveys conducted in Guba region. And we have found the yellowing peach trees and collected leaves, and then you would like to know...
you don't know how common is in the area? I mean, just a few trees were infected or quite common?
Yes. We have found this phytoplasma species only in Guba, and just one private garden, and but all of the peach trees was infected.
What about another prunus species like apricot, cherry? Have you checked also those for this phytoplasma?
We checked. Yes, during our surveys we collected the cherry plum trees, but they was infected with Candidatus phytoplasma solani, not by Candidatus phytoplasma brasiliense.
Just peach then?
Yes.
Okay, thank you.
So there's another question, Barbara.
Thank you, Gulnara. Which tooth types did you find in Hyalestes and in Reptalus?
Sorry?
The tooth types of the insects for Candidatus phytoplasma solani, what did you find in Hyalestes, species and Reptalus?
For the insects, we have not done the we have not amplified tooth, sekigric, and MLP. We are just amplified the stamp gene. So for the insect, I couldn't say. But for the stamp, they you can see two different. They have two different clusters. We have Hyalestes obsolitus and Reptilus noahi, and they clustered between Hyalestes obsolitus and there was two genotypes which distinctly different from each other.
Yeah, thank you.
Okay, is there other questions? Okay, not from online. Okay, so then thank you very much, Gulnara. And we could come.
Okay, now I would like to invite our next speaker, Syed Ali Riza Hosseini, to present important phytoplasma ribosomal subgroups distributed in Iran.
Good morning. In this presentation, I will introduce you the important phytoplasma subgroup in Iran and in Iran. I will skip the introduction to clarify and explain more about the results. We are working on molecular and biological study and reported phytoplasma from other colleagues in Iran. In result, we find 12 phytoplasma subgroup in Iran on different plant species, and the most widespread phytoplasma were peanut fishes boron, clover proliferation, asteriolus, Candatus phonencium group, and group 20 stool bar phytoplasma group as the most widespread phytoplasmas in Iran. You see the peanut fishes boron phytoplasma group as the most widespread phytoplasma in Iran, subgroup B, C, D, V, and Z, and also number of phytoplasma reported with different plant families in Iran. The most important phytoplasma were Candidatus phytoplasma aurentifolia in southern of Iran. The left is Kilaim and the right is Bakrai. And alpha-alpha vicious boron phytoplasma mainly distributed in Iran as a subgroup D. Two subgroup C phytoplasma of alpha-alpha vicious boron were reported from two provinces of Iran that were not any relationship in polyclonal antibody in ELISA test, and different, they transmitted naturally by different vectors, and we consider these two phytoplasma strains belonging to C subgroup as a separate phytoplasma in one subgroup. Sesame phyllodes, the main phytoplasma, this is infecting associated with Sesame phyllodes is subgroup D, and also group 9 and clover proliferation. And Orosius albicinctus and circulifer hematocephalus, two main important vectors of phytoplasma disease in Iran, especially in central and southern provinces of Iran. Tamarix viches boron, associated with peanut fishes boron phytoplasma, asteriolus phytoplasma, and clover proliferation. Squash phyllodes and the phytoplasma associated with cucurbitaceae, mainly infected with, associated with the subgroup D, Punica granatum, pomegranate, and the cucumber phyllodes in the farm and also in the greenhouse cucumber infected with different phytoplasma like subgroup D of peanut fishes boron, clover proliferation, and stool-bar group. Eggplant, virescence, and phyllodes, V subgroup of peanut fishes boron, and calendula sunflower infected with subgroup Z of peanut fishes boron. The second widespread phytoplasma is a clover proliferation group, subgroup A, D, F, and you see the number of phytoplasma reported from Iran and plant families infecting with, associated with this phytoplasma strain. Greenhouse cucumber is associated with clover proliferation, subgroup A phytoplasma, and different cucumber associated phytoplasma associated with cucumber, phyllodes, and yellowing in Iran. Pinus associated with a clover proliferation in Iran, subgroup A, is a new emerging phytoplasma and all distributed in low amount and low disease incidence in all of the country. Tomato big bat. Another group, Aster yellows phytoplasma, the third widespread phytoplasma in Iran, subgroup B, F, R, and S. And you see the plant family, the plant family and number of phytoplasma reported from Iran belonging to Aster yellows, which is vinifera infected with Aster yellow, subgroup B, Eroca sativa, witches boron, big bat, and flower virescence and phyllodes. Jujube infecting with group 6, subgroup A, and Aster yellow subgroup B, and Lolium rigidum associated with F subgroup of Aster yellows. Aquilegia phyllodes, subgroup R of Aster yellows. You see different disease symptoms. PGMP witches boron group is the fourth widespread phytoplasma infected in Iran. Subgroup A, B, C, D, E, H, and I. Number of phytoplasma reported from Iran and plant families belonging to group 9, which is boron disease of almond associated with Candida phytoplasma phoenicium. Peach witches boron associated with this phytoplasma and also another phytoplasma associated with different group. Onoberichis vicifolia, a new subgroup, subgroup I, yellowing and reddening of the leaves. This is a healthy farms. Stool-bar group, the fifth widespread phytoplasma identified in Iran, subgroup A, and number of phytoplasma reported from this group, Fabaceae and Rosaceae, is the most important plant families. Alhaji maurorum infected with stool-bar group and also peanut fishes boron in Iran. Grafen yellows, and other phytoplasma groups reported less than this group, between 1 and 4 subgroups, like X disease, Punica granatum, associated Punica granatum decline, only one family, ash yellows with 3 plant-hold species, apple proliferation with Pyrus domesticus, and some strain related strain not assigned to a ribosomal subgroup, pear decline in northeast of Iran, rice yellow dwarf group in Cyprus, and Bermuda grass white leaf in Cynodon white leaf, and Cassia witches borum, group 29, is a newly emerging and widespread sporadically in different areas of Iran on Pernus persica, Sophora alepicorrides, Convolvulus arvensis. Number of phytoplasma reported from group 29. On Pernus persica, subgroup A, yellowing and decline. Persimmon, Diospirus kaki, infected with the phytoplasma associated with group 29, yellowing and reddening. Convolvulus dwarfing, arvensis, Convolvulus arvensis, witches borum, and dwarfing. And Sophora alepicorrides, dwarfing and yellowing in comparison with healthy plant. And group 3, 30, group Salceda. And the final summary, 12 phytoplasma subgroup reported from different plant families in Iran with different disease incidence and severity. 12 ribosomal group, and also there seems to be increasing. The group including aster yellows, peanut witches borum, 16s are group 3, and until 30. 44 plant family were mostly associated with main disease symptoms of phytoplasmas like phyllodevirescence, proliferation, sterility of flower, yellowing, little leaf decline. And the most widespread phytoplasma belong to peanut fishes, boron, clover proliferation, aster yellows, Candidatus phonecium group, and stoolbark group. Number of infected plant species by each phytoplasma group. You see, this is a phytoplasma groups, and group 2 with 83 case is the first. In aster yellows, subgroup B, in peanut fishes borum, subgroup D, in clover proliferation, subgroup A, in Candidatus phytoplasma phonecium, subgroup C, and in stoolbark group, subgroup A, are the most phytoplasma reported. And other phytoplasma are described between 1 and maximum of 4 cases. And so far, most of the phytoplasma studies in Iran were based on detection and identification of phytoplasma infecting various plant species, and the most important methods employing were biological and molecular study, as well as non-ribosomal gene, mainly PCR and alpha-PCR 16S ribosomal RNA gene were done. During the last day, due to climate change, weather condition, and hot winters, the activity of insect vector of phytoplasma disease has expanded in different parts of the country. The observation of new plant hosts such as tamarisk, pine, cypress, in which insect vectors are less active and feed less, probably due to preference of hosting, indicates a change in the behavior of phytoplasma vector and in the epidemiological complexity of this disease. And phytoplasma disease control strategies in Iran were mainly based on control of insect vector and agronomical practice for preventing disease outbreak, and we need to identify resistant host species and more focused agronomic-based management. Thank you very much for your attention.
Thank you, Sayed, and we have a few minutes for some questions.
Is just a comment, not a question. the presentation was good, thank you, but better for the future, not only for you, to try to use candidatus instead of group. It will be easier for people not working in phytoplasma to follow us. So all these groups are usually having candidatus as well.
Are there any questions from online people? No. Okay, we can move now to the next presentation for Syed Ali Hassan, and it's about occurrence of Candidatus phytoplasma asteris strain associated with onion yellows disease in Iran.
Thank you very much. I present on behalf of my colleague, Dr. Salehi, occurrence of members of Candidatus phytoplasma asteris associated with onion yellows disease in Iran. Formally, allium cepa flower malformation due to aster yellows phytoplasma strain reported from Isfahan province in Iran, but its subgroup was not determined. And in 2022, a survey was conducted in Fars province in southwest of Iran on onion seed production fields for phytoplasma disease. After molecular study and plus analysis, a phytoplasma detected associated with onion yellows. This is an infected onion and also malformation flowers and healthy one, and comparison and plus analysis and phylogenetic analysis show that onion yellows phytoplasma belong to aster yellows phytoplasma subgroup D in Iran. Formally, Eroca sativa, phyllodes, Calyptus, little leaf, Vitis vinifera, little yellowing and reddening, Ziziphus jujube, which is Boroman yellowing, reported as associated with aster yellows phytoplasma subgroup P. This is the first report of Candidatus phytoplasma asteris associated with onion yellows disease in Iran. Aster yellows, I said in the last presentation, are the third widespread phytoplasma identified in Iran with 40 tree plant species from 19 family, and most of the phytoplasma detected in Iran belong to subgroup B. Thank you very much.
Thank you, Syed. Is there any question for him? I want to ask you a question. Regarding the yellowing on the onion, there is a very well-known virus. It's called onion yellow dwarf virus. Onion yellow dwarf virus. Have been these plants tested for the virus too? Because it's somehow causing similar symptoms. The onion yellow dwarf virus.
I didn't check any viruses in onion, but my colleague and virologist colleague working on onion yellows and garlic viruses in Iran, and there are many reports on onion and garlics.
But I suggest testing of such plants to viruses too, because onion yellow dwarf is very well known and it's causing similar symptoms.
Okay.
Thank you. Okay, if there is no question, then we can move to the next speaker. Mahdi Azdabar to present multi-locus sequence analysis of phytoplasmas associated with cucurbitaceous crops in Kerman province of Iran.
Okay. Very good morning, everybody. Now I'm going to present my research work, multi-locus sequence analysis of the phytoplasmas associated with cucurbitaceous crops in Kerman province of Iran. Almost 1/3 of the agricultural products in Iran belongs to the vegetables, and these are the very important agricultural products in Iran. And in case of my province, Kerman province, is ranked as the first for vegetable production among the Iranian province of Iran. We are using the three systems for vegetable cultivation in Iran. The first is the open fields, and we are using the low and high plastic tunnels, and as well as the greenhouses. So during the year, you can find different type of the vegetables in our country, also in my province. We did a survey for phytoplasma disease in vegetables in my province. We did the standard molecular and biological works, and for PCR detection of the phytoplasmas by using the 16S ribosomal RNA gene, as well as the RP and SEC-Y genes for identification of the associated phytoplasmas. So we did analyze the sequences, and we found that the three cucurbitaceous crops, I mean cucumber, zucchini, and cantaloupes, that showing the phyllodes, little leaf, yellowing, fruit malformation, and cracking were observed in the state in Kerman province, and we found that the phytoplasma associated with these diseases belong to group 2, subgroup D. That Dr. Ali Reza said this is the most widespread phytoplasma group in Iran. In the previous research, we found many plants infected by this phytoplasma group. As you see, sesame, tomato, eggplant, and alfalfa. So these are the most important non-cucurbitaceous hosts, and I mean alternative hosts for this phytoplasma important subgroup in Iran. When we did the review the literature, we found that almost 6 distinct ribosomal groups, I mean 1, 2, 3, 6, and 8, and 12 have been reported in association with the 11 cucurbitaceous genus, species, or varieties in the around 14 countries. You can see the world map for the distribution of the different phytoplasma group and subgroups in the different countries. But in case of Iran, group 2 and group 6 was reported in association with the cucurbitaceous crops, and in my province we just only find the group 2 subgroup D. Thank you for your kind attention.
So we have time for one question. Okay. If not, then thank you so much.
Okay, thank you.
And now we move to the next speaker, Apura Shrivastava. She is going to present detection of 16S-R2D phytoplasmas associated with tomato little leaf in eastern Uttar Pradesh.
Good morning, everyone. Hello, salam, namaste. This is Apura Shrivastava from working at the Molecular Plant Pathology Laboratory, Department of Botany, Gorakhpur University. I will be speaking about the detection of 16S-R2D phytoplasmas associated with tomato little leaf in eastern Uttar Pradesh. So tomato, as we all know, is an important agricultural crop loaded with important and active phytochemicals, carotenoids, vitamins, and glycoalkaloids. In the pie chart, you can clearly see that India is the second largest producer of tomato at global stage, and it contributes to 11% of its total production. Now, a number of phytoplasma groups have already been reported to be associated with tomato, but as we all have already heard, the name 16SR2D group always comes up because of the repeated disease incidence. Now, tomato little leaf is an emerging disease in the eastern Uttar Pradesh region. This is the very first report. I would like to show you the symptoms. This is the symptoms we observed at the Rajahi village agricultural fields in Gorakhpur, Uttar Pradesh. Talking about the methodology, we collected crops showing symptoms, and we isolated the DNA using CTAB method. We further went for nested PCR assay using the P1, P7, R16F2N, and R16R2 primer pairs. Then we obtained an amplicon. When we got the sequence, we went for phylogenetic analysis and virtual RFLP. Coming to the results, we observed up to 5% of disease incidence during field surveys. The nested PCR assay yielded amplicon of 1.25 kb size. When we got it sequenced, we did blast and search comparison where it shared 98% sequence similarity with the tomato witch's broom from Egypt. The phylogenetic analysis also showed the clustering of the obtained sequence around 16SR2D group sequences, and the virtual RFLP helped us with subgroup D attribution, as you can see. Coming to the conclusion part, we already have helped us to acknowledge the devastating effect of 16SR2D group, Candidatus phytoplasma orangifolia, in tomatoes. It has been reported from different corners of the world, even here in Oman by Dr. Ali. In India, we have reports from Telangana, Karnataka, and Western Uttar Pradesh. So therefore, it is an alarming issue for us. Talking about Uttar Pradesh, you have already heard that it has been divided into eastern, central, and western province depending on different weather conditions. Now we have reports from western part of the state, but this is the very first report from the eastern part of the state, which shows the disease expansion to new geographical areas. Now, in order to protect our tomato crops, it is important to study the distribution of disease to new areas. We need to focus on the epidemiology involved to adapt well-focused management measures. That is all from my slide. This is my poster. I hope you all visited it. If not, today you can come at the coffee break. I would like to thank the IPWG committee for this opportunity, as it provides great insight and exposure to young researchers like me in the field of phytoplasma. Thank you all for your time and attention.
Thank you, Apura. And we have few minutes for questions.
Just a quick question. Many thanks for your short, clear presentation.
Thank you, madam.
And I just like to clarify the situation of your phytoplasma, because it's not clear, the classification.
Yes, we need more studies.
I hope you will be able to do more, because
we need to
your
cross-verify.
This phytoplasma was one in the list of yesterday, Aurantifolia australazea, is quite interesting. So I suggest you to do more sampling and to cross-verify with more sequences.
Definitely.
Because the only brother is one in Egypt, as you said,
Yes.
and also for this is very unclear situation.
Sure, ma'am. Thank you.
Any more questions? Any more questions? Online? Okay. Then we would like to say thanks for all speakers for being within their time limits, and also to thank all audience for their attention, and thank you so much.
Thank you.
Hello everybody. I think we can start with the section reliable and fast phytoplasma detection methods. And the first contribution is by Barbara Jarosz, and his contribution is development of multi-level monitoring systems for the identification of phytoplasma diseases in German viticulture areas.
Good morning, everybody, and thank you, Franuba, for the introduction. So the title you know now already. I will switch immediately to the presentation. I will present you a current project, and a national project, which is being carried out by a consortium of several partners in Germany from different institutions and with very diverse competences. So the main concern of this project is the definition of monitoring tools for early detection of flavescence dorée phytoplasma. As you all know, grapevine flavescence dorée phytoplasma is classified as quarantine pest in the implementing EU regulation, while Point Noir caused by Candidatus phytoplasma solani has a lower risk potential as the disease is not transmitted from wine to wine. Candidatus phytoplasma solani is therefore classified as regulated non-quarantine pest, as the quality of planting material can be significantly affected by infected propagation material. So far, Germany is regarded as free from FD, but with the entry into force of the new regulation, all EU member states are obliged to conduct risk-based surveys during defined periods to check for the presence of union quarantine pests such as grapevine flavescence dorée phytoplasma. For this project, we collected data in 2021 and 2022 in reference plots in different viticultural areas in Germany to evaluate the current situation of GY, grapevine yellows, in German vineyards. Summarizing the results of these two slides, we have a spread of Point Noir in all viticultural areas in Germany. We have so far no FD sensu stricto, apart from one single wine infected with the FD type M38 in 2020, which has been immediately eradicated. And the non-epidemic variant of FD phytoplasma, the Palatinate grapevine yellows, is so far only present in our region, Rhineland-Palatinate. So based on this situation, we developed a monitoring strategy. So we have the vineyards on one side with the 3 different phytoplasma diseases, which cannot be distinguished by visual symptoms. They have all similar symptoms. On the other side, you have the associated epidemic cycles within which only FD is epidemic. So for the discrimination of the 3 phytoplasma diseases, we choose the spectral analysis in the non-visual range for detection, and for in order to decipher the epidemic cycles, we use risk maps. So bringing together these 2 components, we want to develop a multi-level monitoring system by using drones and quad. I already mentioned so far Germany is free from FD, but what we know from previous presentations and from recent publications, the FD phytoplasma is of Palearctic origin, and alder trees in Europe are the main source for these FD-related phytoplasmas. Furthermore, in Germany, the Nearctic vector Scaphoideus titanus did not yet exist, or we don't have it, but we have alternative Cicadellida vectors living on alder trees, such as Orientus ishidi or Aligus species, which are able to acquire FDA-related phytoplasmas from alder and transmit it occasionally to adjacent orchards. So RLP AgroScience has developed those risk maps for the monitoring. So what you can see here, we have small rivers, and all along these rivers you have the alder trees, so stands of alder trees, and these are the green triangles, and in these red market zones you have the potentially endangered vineyards. So for our sampling, we choose, for our sampling, we choose those adjacent vineyards we choose for our sampling, so the risk zones, and we not only differentiated between phytoplasma diseases, but we also included other biotic and abiotic stress symptoms in the spectral analysis. So we took three red, three white varieties in the selected plots, showing symptoms of GY, virus deficiencies, or damage by leafhoppers, as those diseases may also exhibit similar symptoms. So we took a couple of leaves and samples from symptomatic vines, resulting in 600 leaves per disease and 600 leaves without visual symptoms as healthy controls. So we first made an image acquisition of these single leaves with a hyperspectral camera in the range of 400 to 1000 nanometers in the lab. After this, exactly these leaves were used for DNA or RNA extraction and disease characterization using the standard methods to have a direct correlation between the images and the infection status. So during the next slides, I want to summarize the first results of the image annotation in the visible near-infrared range with NIR. This is a work package which is worked by other colleagues, so I tried to roughly summarize this. I hope it's clear for everybody. So in this mean plot, is plotted the mean spectra for all samples of the red varieties, and each color is represents one of the disease classes. By a mere observation, you can already recognize that we have differences in the curves or in the curses of the wavelength. Just to be a bit clearer or to explain, the mean spectra represents the intensity of all the 224 channels of the used hyperspectral camera plotted over the associated wavelength. And this is a standard procedure to get a first overview of the high-dimensional hyperspectral image data. We did the same thing for the 3 white varieties, so plot for all mean spectra of all sampled white varieties, and here once again the colors correspond to the 6 different disease classes. And we found nearly the same image, so already differences in the curses of the wavelength. To better evaluate these differences, a principal component analysis called PCA was performed. This is a dimensionality reduction method in which the information of the 224 channels corresponding to 224 dimensions is mapped onto 2 dimensions. This is what you see in the two plots or graphs here on the right hand and on the left hand on the right hand. Sorry? So the results of the PCA are depicted separately for 2 sub-areas, the 400 to 700 nanometer range and the 700 to 1000 nanometer range. In this way, the information of the classical RGB range, which is 400 to 700 nanometers, and that of the near-infrared range can be evaluated in a more differentiated manner. So, and what you can already see here, we have nice clusters. Therefore, the results of this annotation looks already promising because we have relatively clear clusters with largely low entanglements here for the different disease clusters or disease classes. And in the 700 to 1000 nanometer range, the data variance appears even slightly larger than in the 400 to 700 nanometer range. The different classes are differently entangled in the 2 ranges, and this is important because it would be a very clear annotation, or a result would be if you have precise clusters without entanglement, and this we are not so bad with that already. So coming from the hyperspectral imaging in the lab to the airborne hyperspectral imaging in the so-called Faunier and sphere range. For this, we are using a drone. Oh, sorry. For this, we are using a drone, and this drone is is equipped with an integrated system composed of a head wall, so-called head wall co-lines sphere sensor, and a high-performance GPS, and a so-called LiDAR software. What does this mean? So this integrated system has sensors which capture hyperspectral data with a very high resolution, and this is important because the individual symptomatic shoots must be identified for data evaluation. The data cannot be recorded closer than 24 meters to the ground, otherwise images will be blurred. The LiDAR measures distances with extreme precision using pulses or laser light by generating a point cloud. And when we combine, when combined with accurate positioning data from a global positioning system, such as GPS, this can be used to produce an extremely accurate digital elevation model, DEM, of the terrain, and this is important as vineyards are often steep slopes or uneven surfaces. So these airborne hyperspectral data needs a post-processing, and for this we need a so-called auto-rectification. What does it mean? The post-processing or the auto-rectification is the merging of the hyperspectral image data and the accurate positioning. As you can see in the raw hyperspectral data, the vine rows here are uneven, or they are more like waves. And here we have a crude tab for the referencing or localization. And this, by this use, the reflectance is correctly calculated. And within the individual pixels, individual pixels, which are very small and difficult to see and therefore cycled, we can already see different spectra of healthy or symptomatic vines as well as ground vegetation. Maybe it's slightly to see, I hope so. These data sets are then further processed by using different software. And for this processing of the software, two different software packages are used, and with this you can have a training of algorithms using machine learning. Excuse me if I cannot really explain all these processes, because this is not my topic, but I try to do it roughly, and if you have questions, I try to answer. So for the software and for the training of the algorithms and the machine learning, the software Mira class, per class Mira we are using, enables the training of algorithms with a hyperspectral image data using the machine learning system. Based on the sample data, automatic disease detection can be achieved in real time. In the labeled image, individual symptomatic pixels so we have here the labeled symptomatic pixels are marked in pink, and the algorithm finds pixels with the same spectrum, which are then also classified as phytoplasma. White and red varieties need to be trained separately. And the ENVI software can be used to identify the wavelength relevant to phytoplasmas and to create a database. If I come to the system realization of a ground-based monitoring using a multispectral camera, so before this was a package which has been done by using a drone, and now we have this ground-based monitoring system with a multispectral camera, and this is installed on a quad. So we have a multispectral camera system, and we need flashlights on this system to obtain homogeneous light conditions, and these flashlights are triggered simultaneously with the multispectral camera. But we also need a very accurate and precise georeferencing and positioning via a GPS system, so that we can build or construct a three-dimensional image of a single vine while the quad is running. I wanted to show you this now in under field conditions. For this, unfortunately, I must escape the presentation models because there is a problem with my video. So here you see, I hope it runs. So the process using the drone, it's just to give you an idea how it works. And this is when we took images with a multispectral camera in the vineyard with a quad. Thank you for your attention.
Thank you very much, Barbara. And is there any question?
Yes, thank you. Thank you, Barbara, for your presentation. I know that it's impossible to consider all the factors, but if you have problem with nematodes or fungus that associated to grapevine trunk disease, the symptoms may interfere with the lectures. Do you have idea? Microphone.
Yes, we know that for example, ESCA disease, these trunk diseases, we also integrated, or we also sampled ESCA-infected wines, but these are only the first results. So far, not all images are annotated. And so first, our colleagues from the institute who is responsible for this image annotation, they first concentrated on the classification of these diseases, which are, okay, at least for us, more important.
The system may discriminate this.
Yeah, but you're right. I mean, we need precise, or we are looking for specific wavelength for phytoplasma detection, and we must be able to distinguish this from all other diseases provoking similar symptoms, and therefore we go step by step, and we use this machine learning system so finally to exclude all mistakes we can have.
How many times the drone is able to cover 1 hectare of vineyard? The drone, how many times needs the drone to cover it to read 1 hectare of vineyard? More or less.
Microphone.
Yeah, let's say 50 minutes. Yes. I mean, it runs raw by raw, and if you have, it depends on the hectare of it. Pardon? Yeah, okay. Maybe 50 minutes. Yeah.
Barbara, just another curiosity. With the system of photograph, because I had students, they would like to do this, but for plant pathology I think it's difficult. The drone or the picture can distinguish the color in the position of the leaves, because the yellow symptoms are in some shape, and if it is, for example, red around, can be insect. So is possible to distinguish this or not now? The position of the symptom of the color change.
Microphone.
It's difficult. You mean if we fly by drone and then we take pictures, if we can not only have a look from above, but also in the from the side, if I really understood. This is possible with this drone, yeah, because it can produce a three-dimensional image, and it can how do you call it? It can I'm not expert for this. Sorry.
So, if it's the last question.
Yeah, my question is kind of a follow-up question of Sultan's question. So you use, when you take a picture, the image, so you use the entire plant or entire entire leaf, or you divide into the segmented, the part and see the individual pixels. So it's really depends on what part you choose. The result is completely different, right? So my question is, which one, what kind of image part you choose, like the whole plant or the one leaf or the segmented leaf part? So it's my question.
So you mean for the For the okay, yeah. So it depends if we take the image with a drone, or if we take the image for the sampling or for the imaging in the lab. But okay, as Wolfgang already told, it's always pixel based. Yeah. Yes.
So thank Barbara for very interesting contribution.
Thank you, Barbara. We would like to invite Wolfgang for two presentations. The first, digital phytoplasmology, remote sensing of fruit tree phytoplasma disease, and leaf reddening as suitable symptoms of pear decline for remote sensing.
So thank you for the introduction, and I will continue with the same topics, but this time I will talk about the remote sensing of fruit tree phytoplasma diseases. Again, as I'm not a digital guy, I have a consortium to do this. It's one Fraunhofer Institute from Germany for cognitive processes and systems, and one company for spatial business integration. And first of all, I would like to introduce or propose a term which I call digital phytoplasmology, because I am more and more involved in collecting, processing, and analyzing a lot of digital data. And we use artificial intelligence, and for this is the so-called machine learning methods. And we should not only apply GPT, but also for phytoplasmology we should apply these artificial intelligence methods. And maybe it's a new subject which we have to deal with. You may know digital PCR, and you certainly know bioinformatics, but this is not what I will talk about. But you heard on Monday systems biology, the temporal network dynamics for Marina Damastia, and this could be already digital phytoplasmology to have data analyzed and new insights. What Barbara presented you is the establishment of risk maps, and this is what we are doing also based on geodata analysis using machine learning methods. The maps she showed you is for the entire country of Rheinland-Palatinate. And what I will talk about today is again remote sensing of phytoplasma diseases by analyzing spectral data, as Barbara showed, for the grapevine, and we use for sure machine learning methods. But I would like to remark, all this is not possible if you have good quality data. So you need what we call ground truth. You need us as phytoplasmologists. And also at the end, you need the phytoplasmologists to judge the data. The background I will more or less skip, because we have deal with pear decline and apple proliferation diseases, which you are well known now at the end of the meeting. And one of the strategies to fight against this disease is still timely uprooting, in particular in young orchards, to prevent further spread of the disease. And this, for apple proliferation, it's even mandatory in some North Italian regions, so we have to have a large-scale monitoring anyway, as for FD in grapevine, and as AP is spread by the vector on a regional scale, it has to be on a regional scale. And these large-scale monitoring strategies for the moment are done by human, and these are not as exact as we would expect it, and our methods could maybe compete if we have achieved a remote sensing based on spectral data. This is the background. The strategy we had to adopt it to identify the symptoms which are applicable for remote sensing, and therefore we choose also these two diseases, apple proliferation and pear decline, because we have a reddening of the trees, partial or entire in apple proliferation, or red colors in pear decline in infected trees. So far it's not considered as typical symptoms, so therefore we had to establish first a correlation with the molecular detection of the pathogen. Then, as Barbara already showed, we made an analysis of the leaves of infected and not by hyperspectral analysis. Try to identify spectral signature which can be used for multispectral purposes. Always we use machine learning techniques. And then we also try remote sensing with drones. And again, this is a different story. If you do this in the lab, you have to, you cannot transfer it to drone data because they have different data. Again, hyperspectral drone data are needed in machine learning to achieve a multispectral drone application. And we will go a step further. We will go to remote sensing with satellite images. And again, there you have multispectral images which you can analyze with machine learning technologies. And to make this reliable, there's another point which is very important. This is the timing of remote sensing. And for this, we studied the induction of the symptoms of the diseases. And regarding the satellite images, you have images of 100, 200 square meters, and you have to find first apple and pear orchards. Again, here we need ground truth. The ground truth is what we stupid guys do. We go out in the field, monitor 20,000 trees and 68 orchards representing 20 different cultivars and for consecutive years. In our study region, this is Palatinate region in southwest Germany. So regarding apple proliferation, we have a big advantage. We have very specific symptoms, as you can see in large steeples and witches' brooms. And they are 100% correlated to our PCR detection of Phytoplasma mali. You see always the numbers what we used. But these symptoms, particularly enlarged steeples, are only visible for experts. So sometimes unskilled people will miss it. And when we did this, we had sometimes only one leaf like this, and the tree was PCR positive. Just to give you an idea that this is a specific symptom, but not easily visible. But these symptoms, these specific symptoms, are really correlated to redding in our study, 96% based on 2,700 data. But the problem is we have a lot of reddened tree which have no specific symptoms, and these we had to test it, more than 500 tests, and we had 82% correlation to the molecular detection of the phytoplasma. And we also important contrast to grapevine, we had no other reasons which we found that induce red colors in apple. So this symptom is very highly correlated to apple proliferation and used for remote sensing. And important is also how this reddening in apple proliferation affected trees is induced. There were already preliminary work done with our colleagues from Trentino. They found that this is induced by cold night and high day temperatures. We repeated these experiments, and this just to go briefly, we use ex vitro plants infected with this strain, like here, and healthy in vitro plants from the susceptible cultivar Golden Delicious. We had two temperature regimes, one 20 degrees at daylight and 5 degrees at dark in a climatic chamber, and as control 22 degree and 15 degree in a climatized greenhouse. And you can see here in the blue line, if we establish a redding index based on intensity and surface coloring, as you can see on the photo here, and after monitoring, regular monitoring of these symptoms, 10 leaves per plant, you can see that the induction of reddening takes place in the cold chamber and not in the greenhouse. So it's really after 14 days approximately, and this was done in the middle of August, so in summer when we do not have any red leaves in nature, there you can induce these symptoms. And in nature this is really in September and 14 days after you have the symptoms, so you can time your remote sensing very precisely. Per decline, I will skip a little bit because I have the next presentation and I have the poster with this kind of data. But again, we have a very good correlation ranging from 75 to 68% of reddened leaf trees correlated with the presence detected by PCR of Candidates Phytoplasma puri. So, in contrast to aplaproliferation, these symptoms are not induced by cold night temperatures, but they appear already at the beginning of August, when it's very hot still. And you can see in September here trees are very nicely. This is the best period. Also, again, in the next presentation, the details. And in October, we have entirely red orchards, sometimes with other reasons. We do the same for the hyperspectral analysis. We have a lab-based system with 2 camera systems ranging from 400 to 1000 nanometer and 950 to 2500 nanometer. The 4 leaves are made on a plate, and then it runs below the camera system, and the spectra are taken. And then you have to see what they asked before, how to analyze this data. So you have these leaves, you have to target what is a leaf, what is a hole in the leaves, what is the background, what's the foreground. And all this is done by an automatic pipeline and also with digital methods, so that we have at the end pixels which cover one leaf area which we want to analyze. And this is the same procedure as Barbara showed you for grapevine. It's a little bit simplified in our case. It's still preliminary data, but to show you the spectra. And here I have to say that these digital analysis guys, they have not so much correlation to the field apparently. So the infected is in blue and the healthy is in red. To my opinion, should be inverse and will be in the next slide. So I keep that in mind, please. So you have here this part where you have the red reflectance, so what you expect from the red leaves, which is higher than the green part of the leaves, and you have this small differences. This is quite important. This is the red at shift. It's something which is usual if you have a plant which is not good health status. So this is not absolutely specific for the phytoplasmas. And you have the same, even nicer with per-decline leaves. What we expected, because this was done also in the range until 2400 nanometer, in the invisible range, that we expected more differences between infected and healthy leaves, but there aren't. But if you calculate the accuracy of these data by AI performance analysis, we have very good data of 97%. But still, it's preliminary and it's not specific for the typical phytoplasma. What you have to do if you apply it for drone, as Barbara also showed, you have a drone with a hyperspectral camera system. You have a lot of different single individual images, and you have to compose one image out of this. So it's a so-called auto-rectification, that you have this image of an orchard. And then you need to train the software, and this is what we are coming into place again. So we monitor one orchard and tell which is the infected tree and which is the healthy tree. And with this data, the machine learning algorithm can analyze the data, so that in red you have the infected tree and green the healthy trees which have been used as control. And again, if you take this spectra from the drone, they look similar to what I have presented before, but they're not so clear, because you have a lot of different variables which interfere from the ground, and the trees are not so clear-cut red and green. So this is much more difficult. But we still try to analyze this in a multispectral drone. We use a commercially available multispectral drone from DJI. And this has 5 channels which are shown here. And if you match it with our spectrum, you see that these channels do not perfectly match our spectra, but nevertheless we try to use it. In future we will probably have drones where you can configure your camera as you like. This discriminant analysis comparing infected and healthy wavelengths just show you that when it's the white color you have a very good discrimination. This is in that range what we already observe. Based on these data you can establish indices and make a health status map. This is shown here for pear and apple. Again, blue is the symptomatic one and red is the asymptomatic trees, and you can see that blue and red are dispersed, but you have also intercalated colors where the analysis is not clear-cut. And this is easier in pear than in apple, because in apple you have symptoms more on the side and only, as I said, partial reddening, so it's more difficult. But we wanted to go a step further to satellite images, and there's, as I said, you have first to identify apple and pear orchards. There is a whole flow chart of processing just to show you what is behind. And again, we had to do the ground truth. Where is in a square of 2 areas of 24 to 24 kilometers apple, and where's pear, where's other orchards and other things, and what does the analysis does with the images. And this is pixel based, this is not orchard based, but you can, if you compared what we see as apple and what the software sees as apple and the images, then it's a very good correlation at the end. But you can see also here they consider some plum as apple. It's not perfect. That's how it looked like. These red regions are apple orchards, and you can see in a satellite image you can deduce trees. And this is a proof of concept for apple. At the beginning of the project, we choose in 2 different orchards clear-cut in symptomatic apple trees, and the dashed line is the control, and you can see that we have similar images as from the lab spectra. It's not so clear in these 2 pictures, but if you deduce it from the different wavelengths, center wavelengths, which the satellite has, and you can see the baseline is the asymptomatic trees as mean, and the individual trees are above this baseline in these wavelengths. And this is from Worldview 3, a satellite which has 8 bands and a resolution of 1.2 meter. So for each tree there are not many pixels which can be analyzed. But everything progresses, and for this pear orchard from last year's, we have Worldview 3 image data which have a resolution of 15 centimeter. So you have a lot of more pixels per tree. And it's just to show you, this is the orchard and this is the image from the satellite. This at the same time the ground truth data. You have green and red trees, and you don't look at the colors. They are not the perfect colors, but you have different colors, as you can see, from green and red trees. And you see also nicely that the greens are more vigorous than the others. And if you analyze these data, and we have 46 symptomatic trees which we tested positive and 46 asymptomatic trees which we tested negative by PCR. If you analyze these data and apply known biomass pigment indices or only pigment indices, so vegetation indices, you may know NDVI, which is very common and which matches quite well our different spectra. And you see here in red the symptomatic trees and green the asymptomatic trees, and the stars show you that there's a statistically significant difference between these two trees types. And also for the pigment indices we have this significance. Unfortunately, this is carotenoid and chlorophyll content. Unfortunately, for the anthocean index, which would be the most important for us, the difference is visible but not statistically significant. But this is a proof of concept that we can detect infected trees in satellite images. And so I'm at the end. The leaf rotting for these 2 diseases is an applicable symptom for remote sensing. The timing is important, and we can distinguish this on the leaf level and the lab on the drone level. and on the satellite level. So thank you for your attention.
Wolfgang, you should continue your poster and then questions.
As you like. So this is just again the basis of the work I presented just before. For per decline, because this is less clear than for apple proliferation. You don't have specific symptoms as for apple proliferation, and you just have this reddening, and you have also problems that 1 year does it appear and 1 year not. But it's again the same problem for the PCR detection. Again, the problem I showed you in the last presentation I had on the psilid vectors from this Candidatus Phytoplasma puri. We have a big problem also with the climate change, that in dry summers the infected trees die. And I already showed you this image from where we know there were infected trees, and next year they are missing. And timely uprooting in young plantation, not in this one, is the only means what we can do. For this large-scale monitoring would also be suitable. And these are the data which are on the poster. In this case, we monitored more than 6,000 trees in up to 13 orchards in that period, which I mentioned before. Tested more than 750 trees by PCR. And what we found, you can see in different years, different numbers of correlation for reddened trees from 75%, 86%, and so on. Every year above 75%. But I have to mention we have also a high number of latent infection in asymptomatic trees. This is a problem for every monitoring. But the period I already mentioned briefly is September is the best. We have even up to 90% in 2020. But we have in per a lot of different variation in colored leaves. So I classified them as green-red leaves, like you can see here, bright red leaves, and complete your entire red leaves, or even sometimes brilliant red leaves. And if we analyze this data according to these leaf colors, we have always a good correlation between every type of color. So the color is not really a good or a better indicator, let's say. A little bit of tendency that these brilliant red colors are better as indication even for normal prospection. And so we analyzed also whether these red colors are correlated to the concentration of the phytoplasma. You can imagine that the green color is not as concentrated as the full red color. For this we tested from 4 known per-decline infected trees, 10 green leaves each and 10 reddish leaves each. And first of all by PCR, and all 40 green leaves we did not detect Phytoplasma puri. And here's an example of one tree, the 10 leaves of this tree with the red colors. You can see the variation, and we detected only in half of them the phytoplasma. And when we applied the quantitative PCR, and this is in this case the phytoplasmas per nanogram DNA extract, you see a variable range of concentration which is not obviously linked to the color. So I would conclude that these different colors are not correlated to the intensity of the phytoplasma in the leaf pestule, which we analyzed. And that means for me that there is an indication of red color that the entire tree is infected and the reaction is somewhere else induced, that you have a red color trees. And so that is the conclusion that this is a reliable indicator for per decline in September is best. And what I did not show is that we tested more than 100 cultivars of per. We tested a collection, and they all show some sort of reddening, and they are all correlated to the phytoplasma presence. So this is really a general symptom for pearl decline which can be used for remote sensing. So thank you for your attention.
One question, please, because we don't have more time. Is there any question? If no, thank you very much, Wolfgang, and we can continue with contribution from Slovenia with Zala Kogek-Zwitter, and the contribution is entitled Unraveling the Puzzle of 16 SR5 phytoplasmas in hazelnuts, a systematic study of sampling and detection.
Okay, hello. I'm coming here from the National Institute of Biology from Slovenia, and I'm going to tell you a bit about the gray clouds storming around our hazelnuts orchards. A bit of background, the hazelnut ranks among 10 most cultivated fruits in Slovenia, and in 2018 we detected 16 SR5 subgroup C phytoplasma in cultivated hazelnuts, and as you can see, symptoms were observed as yellowing, dying of branches or the whole shrub, and also some symptoms were observed also on nuts itself, so there were quantity and quality losses. The molecular analysis of 4 genes showed that they are identical to isolates that are capable of causing grapevine flower seed disease. So this immediately rang bells, as it is the quarantine disease in grapevine. However, it was not the first finding of 16 SR5 phytoplasma on hazelnut. It was already before found in Switzerland. However, it was found in asymptomatic wild hazelnuts in the vicinity of Flavescens dori infected vineyards. To first eyes look to the vectors, so it immediately looked to Orientus ishide. It is a foliphagous species, which is an invasive species in Europe, and it's spreading quite fast after the first detection. And in 2009, it was the first detection of 16 SR5 phytoplasma in this Asian leafhopper. So it was proposed as the vector between hazelnuts in these infected orchards. We did a study sampling not only hazelnuts but also grapevine. These are 3 vineyard regions in Slovenia which were sampled for grapevine, and the points show locations of hazelnut orchards. Some of them were intensive, some extensive, and 3 were from private gardens. 2 of these locations were also sampled for Orientus ishide, and the populations were quite abundant. In just half an hour, we could, by SweepNet, catch like 50 specimens. So it was a really high population. How do we sample? As we all know, the sampling is the first step and crucial step of phytoplasma detection. We were sampling symptomatic shrubs, and they were sampled on the 3 different spots around, so 3 different branches. Leaves were taken and then leaf veins excised. But we also sampled roots on the 3 parts, so you can imagine it's quite tedious digging out the roots. And it also in lab, it needs a bit extra work. Extensive washing is needed for the removal of the soil, which inhibits later molecular analysis. And then the phloem from roots were excised, also 1 gram. We did DNA extraction with our semi-automated Kingfisher procedure, and then did qPCR tests, 18S for the control of isolation, universal phytoplasma test, and the specific 16SR5 group test. The samples that were positive for 16SR5 were also done with nested PCR for map gene, so that we could know the map genotype of these different samples. So electrophoresis and Sanger sequencing was done. If we go to results overview, the survey was done from 2017 to 2021, and altogether 97 samples of hazelnuts were picked. And out of these, 47 were 16SR5 phytoplasma positive. In the first years, like 17, 18, we could detect also Candidatus phytoplasma fragaria and some mixed infections. But in later years, just 16SR5, so the hazelnut decline was associated to this group. If we have an overlook of the orchards, 6 out of 8 intensive orchards were infected, 3 out of 7 extensive, and 1 out of the 3 private gardens. One of the intensive orchards was monitored throughout the 5-year period, and the percentage of infected, of the sampled symptomatic shrubs that were infected with 16SR5 grew from 30% in 2017 to 19% in 2021, even though the grower uprooted the infected and symptomatic shrubs. What did we found with the study of comparing roots and shoots? So 32 samples were sampled in these subsamples of shoots and roots. And if we quickly look at the graph, 80% of samples had a higher concentration of phytoplasma in roots than in shoots. These bigger points represent root samples, and the fuller points are shoot samples. So 16 samples, we couldn't detect the phytoplasma in shoots. However, there were some, especially in 2001, when we sampled in July, that we could detect also in shoots. There were 6 like this. However, there was just one that we couldn't detect in root, but we did in shoots. So testing of roots is for sure more reliable than of leaves and shoot samples. And the ones that are marked with orange were then also we were able to determine the map genotype, and it was always the same in root and shoot sample. Here all the other years it was sampled in October and September. These July samples maybe show that there's some seasonal influence, but a more periodical study would need to be done to say anything concrete. To see a bit which map genotypes we found, just on grapevine there were 3, but M54 was most prevalent, and it was also discovered in the Scaboideus titanus samples. And there were 2 other more minor genotypes, M158, which was discovered for the first time in Slovenia, and M51. Then the next ones were common to grapevine and hazelnut. These are M38, M50, and M122. All these 3 were also detected in the Orientus ishida samples, so which shows that it's really the possible vector between the hazelnuts. The next group is, are the genotypes that were found just on hazelnuts, and the groups marked with, the genotypes marked with yellow are the ones that were detected for the first time in Slovenia, and also it was described for the first time. So these are 4 that were found in hazelnuts, but it was in minor percentages. The ones marked with red are all map genotypes that belong to one of the 3 map FD clusters, so isolates can be transmitted with Scaboideus titanus and can potentially cause epidemics on grapevine. However, here is one, this M48, which is not compatible to Scaboideus titanus and Orientus, so this shows that there are for sure some other vectors. If we just quickly look at the locations. So M54 is present in all regions in Grapevine, and also M38 in yellow. Hazelnut orchards are marked with this hazelnut sign, and it's interesting that the diversity of genotypes was just in one hazelnut orchard higher than in all the vineyards of Slovenia. The conclusions are that testing of hazelnut roots is more reliable than of shoots. The detected map genotypes were the same in these subsamples. 3 map genotypes were common to grapevine and hazelnut. As other studies show, M54 seems to be restricted and adapted to the grapevine's Qaboos titanus pathogesystem. Genotypes that can cause epidemics on grapevine are also found in hazelnuts, and the genotypes found in hazelnuts and grapevine were also detected in Orientus ishide, so it is the possible vector of 16SR5 phytoplasma in these hazelnut orchards. However, for sure our work is not done. Transmission assay are being started this year with Orientus ishide between hazelnut and grapevine hazelnut, and survey of other vectors is being done in these orchards. Sampling of wild hazelnuts and also other surrounding reservoir plants, as we know, which is really important in the vicinity of the infected vineyards, is being done. And we are working on the genome of phytoplasma from hazelnut to be compared on the whole genome level to the Flavarsin story genome. So thank you to our financiers, the ministry and research agency. These are 3 projects which are connected to this work, so you can check more on their websites. I also thank my colleagues from INRA. A part of work was also done at their lab in Bordeaux, and thank you for your attention.
Thank you very much, Salah. And there is one question, please.
Yes. I'm here. Hi. You see the main symptoms in hazelnut is yellowing. Okay. And the complete identification of the phytoplasma is the ribosomal group 5, and you have more information?
It's group 5, C subgroup.
Ah, C.
Yes.
Sorry, I didn't hear. Sorry.
So we have another question there. Kadrian.
Thank you for your interesting presentation. I was wondering, you know, the spreading of the disease in a few years are very high. So do you think that it is only due to the vectors, or there is another factor for this fast spreading?
Yeah, I think it's mostly to the vectors. We really take care of the planting material. But yeah, it's the Qaboos titanus is really widespread, and in the last year, growers have a lot of problems with these regulations of the treatments they can use, and they are quite desperate. So in one part there are big outbreaks, and they don't know anymore how to control it.
But what about the roots? I mean, they look like a very good inoculum source, the roots. What about the root bridging?
Root bridging is one option, but for now, we think it's not really the source. So more of the outside reservoir plants, vectors coming in the hazelnut orchards.
Okay, thank you.
So another question from Barbara.
Yeah, this is what I would like to know. Did you find only adults of Orientus ishide on hazelnut, or eggs as well?
For now, we were just sampling in July, and we found just the adults. We weren't really looking, but yeah, we will in the future.
And the latest question from, I don't know the name, please tell us.
Aziz. Hi, Zala.
Hi.
Thank you for your presentations. I have questions. You mentioned you took a sample from the leaves and roots. So how old the plant that you took sample from it?
Yeah, it's the most infected orchard was like 5 years, but they are replanting all the time, so different year of the plant.
So according to your result, is that age of the plant could affected that the spread of phytoplasma?
Sorry, I didn't get that.
According to your result, is that age could influence the phytoplasma spread or show symptoms?
Yeah, I think maybe later it doesn't really show in the first year, so maybe later. Because this orchard that was monitored throughout the 5 years, the grower really put all his money in this, and then when the bushes should give him the most, they were declining.
So thank you, Zala, and also for discussion. And we can move to another contribution.
Exploring practical application of metabarcoding with Minion to support the surveillance of the following bacteria: Candidatus phytoplasma and Candidatus liberibacter, by Ellen Everest.
So hi everyone. Today I would like to introduce you to the project outline of MetaMinsurf. MetaMinsurf focuses on the detection and identification of phloem bacteria through benign metabarcoding. It's a new project which only started a couple of months ago. So I think you are all familiar with the problem. So the detection and identification of Liberibacter and Phytoplasma phloem bacteria is anything but straightforward. There are many reasons for that. I listed a few on the slide. So first of all, the existence of many species, strains, as well as RFLP groups and subgroups within the taxon. Secondly, because of the simultaneous occurrence of both species with variable and similar symptoms. And thirdly, because often multiple consecutive tests are required for both reliable detection and correct identification. To overcome these challenges, we would like to investigate the potential of untargeted benign metabarcoding using multi-locus sequencing approach, where we would like to combine broad detection and identification of Phytoplasma and Liberibacter in one test. So the first step in our project is to select suitable barcodes and primers and to establish reference databases for both Phytoplasma and Liberibacter. This will be accomplished by literature reviewing and screening, as well as in silico analyses. So the reference databases are established by selecting relevant sequences from publicly available repositories, then by conducting quality control screenings, and ultimately by aligning them. The barcode selection is based on a literature review on the available sequence data, as well as on newly developed identity matrices, for which you can see an example of one on the slide. This is the full-length 16S identity matrix of Phytoplasma, where you can see where the darker red colors signify more sequence diversity among species. And then for the selected barcodes, primers will be selected from either literature or they will be developed by ourselves. So I think nobody's waiting for another pair of 16S primers, but for the other loci, we will explore whether we will need to develop an additional set of primers. Then in the second phase, The first step will be barcode PCR optimization, and then later on we will optimize the Menayan library preparation and the bioinformatics workflow. Because I only have 3 minutes, I won't be able to explain all of this. But in the end, we would like, in best case scenario, we would like to obtain protocols and bioinformatic workflows to detect and identify Liberibacter and Phytoplasma phloem bacteria using Menayan metabarcoding. And we would also like to write general guidelines for transferability to other barcode or organism combinations, because next to the phloem bacteria case scenario, we are also working on forest fungal pathogens. Thank you very much for your attention. But I would also like to take this opportunity to ask you all for advice, especially on the barcode and primer selection. So in case any of you would have suggestions, I would like to discuss this during the coffee or during lunch. Thank you very much.
Thank you very much. We'll continue. Next presentation is validation of modified quantitative PCR and synthetic DNA G blocks control for detection of candidate Phytoplasma mali by Jared Yashwara Bell.
Thank you for the introduction. To save time, I'm going to skip through the title of my talk. You can read it or look in the program. So I'll be talking about Candidatus Phytoplasma mali, which is the group 10A responsible for apple proliferation. It's currently not present in the United States, and as far as to my knowledge, nor North America. But regardless, it is a quarantine pathogen in the United States as well as Canada. But there is always the risk of entry through import of infected propagation material. So there is definitely a need for a validated screening tool for this pathogen. So in our lab, like I said in the panel, basically we will design from scratch diagnostic assays, but to save time, if there are published assays, we'll investigate those and validate those. So this is the case with Phytoplasma mali. I scoured the literature. I selected 3 different assays to evaluate. These targeted 3 different target genes. After some preliminary in silico and wet lab testing, I found that the 16S and ITS, which is the one used by EPO, it did cross-react with other group 10 phytoplasmas, the Prunorum and the Piri. So the immunodominant protein assay showed to be highly specific, so that's the one I selected for full validation. Currently, our screening assay is a general universal phytoplasma assay. It's a duplex of a 23S assay by Hodgetts et al., and we duplex it with an 18S internal plant host from Christensen, so that is our standard universal detection assay. So I duplexed the IMP with the same 18S internal plant control, optimized it, and then did a comparison with our current screening assay. So I ran a battery of validation testing. I looked at linearity, repeatability, determined the limit of detection. For this, I used serial dilutions of the mali DNA in apple DNA, and I used 3 different isolates of mali. I did a specificity and selectivity screen. I also incorporated multiple operators to look at precision, and then I also tested it on 2 instruments, the ABI Quant Studio 5, which is our primary instrument, and then also the Biorad CFX. So I'm going to throw a lot of data at you, but basically for repeatability, there was good repeatability between replicates as seen by the low standard deviations. As I said, I did a direct comparison between the IMP duplex and our 23S duplex. For each corresponding dilution, you can see very similar CTs throughout all the dilutions, and this trend held true for testing all 3 isolates. So if viewed graphically in linearity curves, you can see that the IMP assay and the 23S assay had linearity curves that pretty much overlap, showing very comparable performance. And then for precision, as I said, I included additional operators. So when accounting for the data between 3 different operators and using different ABI Quant Studio 5s, once again, you see good repeatability in the data as seen with the low standard deviations. And again, CT values between the 23S and the IMP assay were very comparable for corresponding dilutions. For all the data, the average variance was about 1% for both assays. And then the sensitivity was determined to be the same for both the IMP duplex and the 23S assay. And at this dilution, which was kind of near the limit of detection, we were able to do conventional PCR and sequence the products to get down to species identification. So for specificity and selectivity, I was able to get various isolates of Mali. So I tested 5 isolates of Mali, as well as some other group 10 phytoplasmas, including Piri and Prunorum, and then just a few other random other phytoplasmas. And during this screen, only the Mali isolates were detected by this assay, and there was no late cross-reaction seen with any of the other phytoplasmas tested. And for diagnostic specificity, I selected 29 diagnostic samples that we had in our freezer. This was from testing that occurred across the United States. So these samples were screen positive with the 23S assay, and then we did conventional PCR and sequencing to get down to species. So in this, we had 2 Piri isolates, and then we also had 4 samples from apple that were 16SR5 group phytoplasma. So this also expressed an additional need for this assay, as screening apple and finding phytoplasma is definitely not indicative of it being Mali. So when testing all of these previous samples, they were all negative by the IMP assay, again showing high specificity for the Mali pathogen. And in another selectivity screen, I also tested 4 healthy host plants, plant tissue, 16 different pathogens of apple and other fruit, as well as 1 commercial biocontrol agent, which comprised 32 unique isolates. And again, none of these cross-reacted with the IMP assay. So I went back and revalidated it to a lesser extent in the Biorad CFX machine. So again, linearity curves more or less overlap, again showing good comparability within this machine as well. And then again, good repeatability, very low standard deviations for both assays in the Biorad. Again, similar CT values at corresponding dilutions. And then these values in the Biorad were also very similar to those produced by the Quant Studio 5. And then in the Biorad, the sensitivity remained unchanged. So I ran a quick check just to double check the specificity in the Biorad, just to see if anything weird was happening. So I tested 3 isolates of Mali, just 1 Prunorum, 1 Piri. I tested healthy apple, healthy pear tissue, as well as some fire blight, and only the Mali was detectable by this assay. So just to summarize everything, so I duplexed the IMP assay with the 18S, and then I validated it in both the ABI Quant Studio 5 as well as the Biorad CFX. The assay performed well for all validation metrics that were tested, and it showed 100% specificity for Mali. The data produced for the Quant Studio 5 was very similar to that for the Biorad CFX, and the data for the IMP assay was very similar to that produced by our universal screening assay with the 23S. We did a cost analysis for this, for the assay. Not sure what that is in euros, but it basically comes out to 92 cents, or less than $1 per reaction. So just like any assay, especially those used for regulatory testing, proper controls are needed with every run. As we do not have Mali in the United States, availability of source material is difficult to get and very limited. And even though we have DNA in our lab, it's unfeasible for us to provide validated positive controls for all our screening labs. So we decided to make a synthetic G-block positive control to distribute, or that they can order themselves actually. So basically we started with a 16S base that has all the primer sites for conventional PCR, and then we inserted the targets for the 18S, the 23S, and as well as the IMP into this target to ensure that we are able to assess whether or not we are contaminating any samples with this positive control. We inserted, aside from sequencing this and seeing an unusual pattern due to having all the having all these inserts in the control, just upstream of the 23 assay, we altered the nucleotide sequence so that when you translate the sequence, it'll read APHIS science. So you'll be able to tell if any of your samples that come up positive is just due to cross-contamination with the control. So I ran this through very similar validation tests, and it showed to work very well. So overall conclusion, so the new duplex IMP assay does improve on the original published assay in that it includes a plant internal control, so you can have quality control assessment for each reaction. When I optimize the assay, I optimize it so that it runs under the exact same parameters as the 23S assay. So while you can run the general screening assay, and if something pops up in Apple, you can run the Mali to double check. You have the ability to run both assays in parallel. So in one run, you can get both general detection of phytoplasma and discrimination for Mali at the same time. So we did draft protocols for both the QuantStudio 5 and CFX, and they're available if upon request. I think I put most of the information in the abstract. And then the synthetic control was shown to be work very well, and now it works with all three of our validated assays, the 23S, the IMP, as well as conventional PCR and sequencing. And then we also do have a protocol available that provides the information to order it as well as prepare the control for use. I'd like to thank those who provided me with DNA for phytoplasmas and other pathogens, as well as healthy tissue DNA. I wouldn't have been able to do all of these selectivity testing without it. I'd also like to thank the committee, the organizers, Sultan Qaboos University, and the host country, Oman, for having us here. These are references if you care, and any questions?
We have time only for one question, please.
One question, just to avoid the problem with proliferation in the United States. Do you know the time where the control you used were collected and used, extracted? Because in Europe we have at least 3 strains, 2 main strains, and the first one detected is called AP15 or AP, is old strain, not detectable anymore. And I am afraid if you use this, using this, okay, you have also the 16S, this can back up the story. But it is correct to evaluate your system also with the newer strain. That was the one making big outbreak in Trentino, Alto Adige, and other regions in Europe.
Yeah, good point. Those are all the ones I could acquire, so that's what I was able to test it on. I did go, the initial in silico analysis, I tested it on basically everything that I found in GenBank, so I'm not sure if the IMP genes are in there from those particular isolates, but I think there was about 33 to 37 in there, and it was basically 100% match. There's 2 SNPs in the reverse primer, but they're accounted for with the degeneracy of the primer, so I didn't see anything in GenBank that would have caused any issues, so, but if we get our hands on the material, it would be great to test for sure.
Just one quick question. The synthetic G-block DNA that you use is a good idea when material is not available. But my question is, did you spike the Apple genomic DNA with your synthetic DNA? Or did you use the synthetic DNA alone as a positive control?
Sorry, did I spike the
apple DNA with what?
Your synthetic DNA that you have created.
The synthetic DNA? No. So the synthetic control has the 18S on it already, so we didn't need to. We put the 18S in the control so we wouldn't have to spike it because that's just an extra preparation step. But no, we did not test it in apple DNA.
Yeah, we can discuss this later on over the lunch, but I think it is important to have the apple genomic DNA spike with your synthetic DNA for the better control.
Okay. Yeah.
But we'll talk about that.
Thank you, Jared.
And we can move to Italy. And the contribution is by Camilla Barbieri, Phytoplasmas in Papaya Detection and Identification.
So good morning to everyone. I will talk about detection of phytoplasmas in a crop that has been mentioned rapidly this morning, the first presentation of Prof. Fiore, that is papaya. So papaya is a very popular fruit tree that is widely cultivated in tropical and subtropical areas around the world, and the major producer countries are located in Asia and America. Unfortunately, papaya is affected by several diseases that have been associated to phytoplasma's presence. These diseases are named differently based on the severity and peculiarity of symptoms, distribution, and the type of phytoplasma detected in papaya's plants. So some examples of these names are papaya bunchy top, papaya dieback, yellow crinkle, and mosaic. The most common symptoms displayed are proliferation and shorter internodes of apex stems with a bunchy top appearance of the top of the plant, intervenial mosaic and chlorosis on leaves they can also present a crinkling aspect. And then we have a reduced production of flowers and fruits, and also apical necrosis that can progress to the death of the entire plant. These diseases can be so severe and can cause 100% losses in some plantation. So these papaya diseases are present almost in all the area where papaya is cultivated. As you can see in this map, we have lots of different phytoplasma detected in papaya's plants, and the most common and widespread are belonging to group 1, Candidatus phytoplasma asteris, group 2, Candidatus phytoplasma australasia and aurantifolia, and group 12, Candidatus phytoplasma solani and australiense. So focusing on our case study, in the summer of 2022, a disease referable to papaya bunchy top has been observed in a trial plantation that is located in north of Italy near Bologna, as you can see in this map. This cultivation was an agronomic trial to evaluate the papaya cultivation under greenhouse condition in Italy, so the feasibility of the cultivate papaya in Italy. So after a few months from the start of the plantation, some plants start to show yellowing symptoms on leaves, production of small fruits that also have premature falling. So we decide to perform phytoplasma detection using both molecular and biological method. Starting from molecular analysis, we extract the total DNA with chloroform phenol method from 1 gram of midribs and lamina tissue from both symptomatic and asymptomatic plants. Then we perform PCR on the 16S ribosomal gene using phytoplasma gene using these primers pair listed in the table, and then we perform RFLP analysis with TRU1 first restriction enzyme and the sequencing on the amplicons obtained. Here are some results. In the, all the symptomatic samples resulted positive in phytoplasma 16SR amplification, while the samples collected from symptomless plants resulted all negative. We didn't notice any difference with the detection starting from lamina and midribs from the same plant, so we obtained the same results. And you can see in the picture on the right the restriction profile on the amplicons. All the samples presented the same profiles, that is belonging to 16SR group 12, subgroup A. From the sequencing, we obtain sequences of about 1,200 base pair that has an identity of 29.67% with Candidatus phytoplasma solani reference strain with 100% coverage on the sequence. So we decide to perform a multi-gene characterization of this papaya Stolbur strain, and we amplified some non-ribosomal genes with the primers pair listed here in this table that are STAMP, SEC-Y, WMP1, and TAF. Okay, so those are the RFLP analyses on these amplicons that have been digested with different restriction enzyme, depending on the more appropriate for each gene. In the picture on the left, there are TAF amplicons digested with HPA second enzyme, and we obtained the same restriction profile for all the samples that were also identical to positive control staph, that is a Stolbur strain detected in tomato in France. In the picture in the middle, there are SEC-Y amplicons digested with TRU1 first enzyme that also have the same profiles for every sample and for the positive control. And on the right, unlike the other gene, the WMP1 amplicons digested with RSA first enzyme showed variability among the profiles of the samples that were also differing to the positive control profile. Regarding the sequencing, the TAF gene sequences had 100% identity with the Candidatus phytoplasma solani reference strain and resulted to be TAF type B1. For SEC-Y gene, the identity was about 99.59% with the reference strain, while STAMP gene sequences had identity higher than 97% between the samples, and they resulted all enclosed in the STAMP variant ST10. For WMP1, the identity percentage between samples were lower than the other gene, and that reflects the variability that we see also in the restriction profiles. And we suppose that this variability maybe can be due to different insect transmission, as a variable membrane protein is involved in the phytoplasma insect interaction. So after molecular analysis, we decide to perform a biological method to evaluate if isolation could be appropriate to detect phytoplasma in papaya tissues. Sorry. And we use the procedure that was published in a previous study of 2016 by Contaldo et al. Here a short video of the isolation procedure. We start from leaves, stems, and fruits of papaya that were also detected with molecular method. So after sterilizing the surface of the tissue, we slice a little amount of the tissue and cut it in a little pieces. Then we put inside the CB liquid medium, of which you can see the composition in the table below. And we incubate the liquid culture. So we waited for color change of the culture and the increasing of turbidity, and then we plate an aliquot on CB solid medium that has the same composition more or less, and we incubate in a microaerophilic condition at 25 degrees. So after 24-48 hours, we obtained colonies that were also observed under microscope, and we purified the colonies with the different morphologies. Okay, here some results from the isolation procedure. In the picture on the left, there are tubes with liquid culture with a different level of color change after the bacterial growth. Under these, there are colonies on the solid medium after 48 hours of growth. And on the right, there are some colonies that present different morphology observed at the microscope at 40 pair magnification. So we perform molecular analysis on these colonies to detect phytoplasmas. And we picked a single colony, heated in water for 10 minutes, and then freezed on ice for 5 minutes. Then we use this primers pair for PCR to amplify 16SR gene for phytoplasmas and bacteria. And I want just to highlight the last primer pair that it's GPO3F MGSO, designed in 2005 for mycoplasma detection. but also proved to detect phytoplasma also in quantitative PCR. And then we performed RFLP on the amplicons and the sequencing. Here are some results. So from the amplicons obtained with the common primer pair used for phytoplasma and the universal bacteria primer, we obtain so often a specific amplification. As you can see in the left, in the agarose gel, some samples present multiple bands, and also amplicons, we obtain amplicons with unexpected size. So from the RFLP, in the picture on the right, we obtain profile that were not referable to phytoplasma swans. So from the sequencing, we could identify these organisms to be bacteria from the genus Pantoea and Enterococcus that are two common endophytic bacteria. And sorry, here you can see some picture of the colonies. From the GPO3F MGSO primer pair, we usually obtain amplicons with the expected size that more or less 200 base pair. And then we perform RFLP analysis. On the picture on the right, there are colonies profiles on the right of the marker that have been compared to profiles of positive control strain. In the colonies profile, I highlighted in orange boxes the pattern of phytoplasma that we could recognize inside these samples, but they also present extra bands and background noise that could suggest to the presence of other microorganisms. So this is the first identification of Candidatus phytoplasma solani in plant papaya in Italy. This result is in accordance with previous study that report stolbur in other cultivation of papaya around the world. Candidatus phytoplasma solani is a widespread phytoplasma in Italy and in many countries of Europe, and it has a wide range of plant hosts. So we can add papaya on the long list of plant hosts for stolbur in Europe. This is also the first isolation trial performed successfully from papaya's tissue, and we could obtain colonies containing Candidatus phytoplasma solani together with other microorganisms, endophytic bacteria. Sorry, I go back to the last slide because I didn't mention that from the amplicons we obtained, from these amplicons we obtained also short sequences that had a high level of identity with Candidatus phytoplasma solani. So the perspective of our work will be to improve the isolation procedure and to formulate a more selective medium for, that could allow the isolation of phytoplasma in poor culture and maybe could allow to fulfill the Cox postulate. So thank you for your attention.
Thank you very much for interesting contribution, including cultivation of phytoplasmas. This is very tricky, I think.
Different topic.
And is there any question, please? Kadrian?
Thank you for your interesting presentation. Regarding your in vitro culturing process, I mean, you just cut into small pieces the leaves and leave in the culture, in the media, and without filtering, you directly transfer to the culture. Is that right?
Yeah, sorry.
Okay. I mean, if I understood right, why don't you crush the leaves? First of all, my question is that. And second one, why don't you making a cold filtering? I mean, filtering the extract through, let's say, 0.45 micron filters, just to avoid contamination.
So for the first question, yeah, we slice the tissue in little pieces, but with the sterile blade, we'd also crushed a little bit to let the lymph going out in the liquid media. And before performing this, we sterilize the surface of the tissue with, so we avoid the contamination from surface.
But it's not
and
enough.
yeah, for the filter sterilization of the filtering, we perform in other trials this step, and usually also we, when we transfer on solid medium, when we see a lot of different morphology of colonies, we go back to the liquid medium and we filter it and then plate again to see if the filter can clean the amount of different microorganisms that then growth in the medium. Don't know if I'm clear.
Yeah, you are doing that.
In papaya, we didn't do this, but
very high.
yeah, in some cases we also do filtering with 0.2 millimeters filters. Yeah.
May I have one more comment? You know, I mean, I have a great experience on culturing Spiroplasma citri from the plants. I know phytoplasmas are more difficult, but maybe in the first time when you crush the tissue, you can directly dilute your extract and then make a dilution serial with the culturing media, because in some of the plants, as you know, the phenolics are very high.
Yeah, these are all procedures that we use and we try in different, the purification of the colonies will be also from plates, solid medium, but also with serial dilution of the liquid medium.
Okay, okay, good luck.
So thank you for the advice.
Just a comment, Camilla was completely right in explaining to you, Kadri, the situation. Usually, when we start the isolation from plant material, as Camilla said, we start cleaning and sterilizing the surface, and we crush the midribs in the liquid medium, and we wait for a few hours, usually, to check the turbidity of the liquid medium. Sometimes it needs to do the dilution. So we do the several dilution before the 24 hours, but we never use at the beginning the filtering because it depends on the plant material. I mean, sometimes we got some turbidity, sometimes we got nothing from the same plants. So it depends really on the case. So we are evaluating time by time the more correct procedure. But we use several dilutions also before plating, and we use filtering with 0.45 micrometer, to at least avoid the majority of the bacteria. Yeah, thank you.
It's a great achievement. Thank you for the presentation. And my question about the collecting the colonies, and are you picking up individual colonies in the agar and then crushing them, or you just collect the whole colonies in some kind of buffer and screening for them?
Actually, we tried different type of picking colonies, and now we are performing the single colonies, heating it in water that we use directly for the PCR mix production, heat and freezing, so shock, thermical shock to let the DNA to be ready for amplification. And this is the best way to obtain right amplicons, because when we pick many colonies, sometimes there is a lot of background noise, so it's difficult then to see profiles in RFLP, and also we use Sanger sequencing, so also to have a good sequence quality. Yeah, one column.
So thank you very much, Camilla.
Okay,
and
thank you.
Next presentation, multigenic analysis for identification of Phytoplasma strain infecting Dimorphandera, Graneriana, and Ternera ulmifolia in Brazil by Svante Bertessin.
Okay, thank you. I am presenting this on behalf of our colleague from Brazil. She could not come. And also is something to show you, again, as I promised yesterday, the limit of molecular detection of phytoplasmas. These two species are from Brazil. Dimorphandra gardneriana is these three you see here, and below you see the symptom in two places in Brazil. This is infected by the group in the past we detected as 16S13 group, and these are the molecular identification carried out by RFLP analysis on these samples, just by specific restriction enzyme. And also the second species infected is Tournera ulmifolia. You see here the symptom in the middle and below. And both these plants are important for medicinal purposes and more. And while the first one was from the upper part of Brazil, this one is just near, I think, Rio de Janeiro. Sorry, but my geography of Brazil is not the best. This is the identification, and also this result to be group 13. We try to do more identification in the past. Some of you following this maybe remember these two plants infected were already presented in previous IPWG meetings. So we try to really verify what was this group 13, but it was complicated. We went to another gene, for example, on RP gene. We found profiles that are not referable to any other published profile. But there are very little study on this phytoplasma that together, Nicola explained it to you beginning today, is, I think, only found in South America, no, in America, let's say, because group 13 is starting from Mexico, as far as I know. In reality, if you look at the classification, in the group 13, we have now two candidatus phytoplasma. One is 13A, candidatus phytoplasma hispanicum, and the second one is 13G, candidatus phytoplasma melie. Both published in the same year by two different groups, one in USA and one in Argentina. So they have very little difference on the 16S. Therefore, what we did was we took these two samples from Brazil in order to know group 13, which candidatus was referable to, if they were the same, or not. First, we did our RFLP analysis on three genes, CK, RP, TAF, and LEO. And as you can see here, unfortunately, the candidatus Phytoplasma hispanicum is not, I think, anymore available as DNA. So we use it for comparison only on molecular data already published. While CATH is candidatus Phytoplasma melie, kindly provided to the collection from Luis Conchi in Brazil, who his group described this. So as you can see, the CHTY is the candidatus melie. By RFLP analysis in the gene, you can see there are some differences. I will not bother you at this time of the day, maybe you look for lunch, but you can see that the two phytoplasma are differing between them, and also are differing with the candidatus melie, just by picture. And by restriction, this is the picture. You see the candidatus melie is the last, and the other are Tournera, the other one. And as you can see, the profile in one case is different on TAF gene, is identical on RP, is identical on CK, is identical on LEO gene, isoleucine. So I this was still difficult. We didn't found the sufficient differences to see what is these two phytoplasmas. We try to follow the revision guideline I showed you yesterday. So we went to the sequencing of the other housekeeping gene, and these are the result of the sequencing. We use the sequence available in GenBank for Candidatus phytoplasma hispanicum. You see in bold the melie, and what is clear is the first one on the left is 16S. Candidatus, the Dimorphanda gardneriana strain has 99.36 and 99 to both. And the Turnera is 99.45 and 99. So on 16S, they can be both, Hispanicum or Melie. So question mark. Then we went to the other gene, and we found the number you find here with the one we were able to amplify. For Turnera, we were not able to amplify the RP. In any case, if you look at the numbers where there are both, the difference is, in some cases, like RP, is good, 96.43 to Hispanicum for Gardneriana and 94 to 44 to Melie. The other is not available. But I will not bother you. Also in this other gene, the situation is not helpful. So again, following the guideline, we could not differentiate the candidatus on the 16S. We could not help support the 16S with enough data on the other gene. This is a kind of summary of this situation. The 13 is very likely an American phytoplasma. It was found from Mexico to down. And if you look at the Hispanicum compared to the Melie in Argentina, the difference is not relevant for the rule we have now, because the Melie is 98.91% to the Hispanicum. And the Dimorphanda and the Turnera are having slightly progressive differentiation on the 16S. 99.36, the northern, and 99.45, the southern, to the Hispanicum. And maybe it's difficult to read the red. You see that to Melie, the difference is, it is exactly the same, 99 in both cases. So what is this? This is one of the, I think, several cases in which, at least with what we have available for molecular classification, is so tricky that is not allowing us to discriminate among candidatus. And this can be relevant for several issues, especially for quarantine, but also for epidemiology. It will make it difficult. It's similar in another sense to the story of the two candidatus, Australasia and Aurantifolia, I showed you yesterday. And I leave you with this question mark, but I think the solution is maybe full sequencing, but maybe also culturing, because culturing can give us all the biochemical, the other characteristic that can help us to differentiate. Very likely, Hispanicum and Melie are the same candidatus, and maybe we can merge the two names and make just one candidatus. But this is for further clarification in the taxonomy. Thank you for your attention, and this is Elena Montano for you. I think you remember her, she was with us all the time, except this, because too distance. Thank you.
Thank you very much, Assunta. And is there any question, please? If no, thank you. And the latest contribution is online, probably. Is Prabha with us? Yes? Again, yes. We can start with online contribution. Fabio is there, please.
Hello everyone. I am Dr. Prabha K, Senior Scientist Plant Pathology from Indian Council of Agriculture Research, Directorate of Floriculture Research, Pune, India. I welcome you all to my presentation. First of all, I would like to thank the organizers of 5th International Phytoplasmologist Working Group for giving me this opportunity to virtually present in front of you. I am going to present the work we have done to identify volatile signatures of Phytoplasma infection from marigold plants showing witch's broom and their potential as biomarkers for diagnostics. You all know about the marigold plant. Marigold is a very common plant in India, cultivated for its loose purpose almost in every flower growing areas in the covering most of the states in India, and it is part of each and every function and festivals in India, and almost 600 million tons of flowers are produced per year in this crop. A major constraint in marigold cultivation is Phytoplasma infection. You can see here the manifestation of Phytoplasma infection in the marigold plants, where this is the healthy flower, whereas the flowers are malformed and causes phyllodes, virescence, and the plants also shows little leaf and witch's broom. The economic part here in the crop is flowers, and when they are malformed, farmers are facing a lot of loss due to the infection. Almost 30 to 40% loss is reported for Phytoplasma due to Phytoplasma infection in marigold. And a vector which is spreading the disease from healthy infected to healthy plants has been identified as Hishimonas fasciitis. To characterize this Phytoplasma, which is causing phyllodes symptoms in marigold, we have done the molecular studies using 16S rRNA sequencing as well as multilocus sequence typing using TUF, SEC-A genes and IMP membrane protein gene. And we could identify the causal organism as candidate Phytoplasma asterisk group, 16SR group 1B Phytoplasma. And in the case of Phytoplasma infection, the marigold, the Phytoplasma infection causes no viable seeds because you have already seen the malformations. Here you can see there are no viable seed production. So in the case infection is a problem in seed propagated plants, as there is no further propagation happens when Phytoplasma is infected. A major variety of marigold cultivated in India is Calcutta type, and it is propagated through cuttings, and you can see if the mother stalk of the plant is infected, definitely the cuttings collected from the plants will be infected by Phytoplasma, and the symptoms are expressed in a later stage of the plant during the flowering stage. So the identification at an earlier stage is failed, and farmer, after giving all the inputs, they have to remove the plant in the flowering stage and they are not getting good flowers. Another problem of the identification of Phytoplasma infection in the field is the mixed infection with viruses and other nutrient deficiencies, often confused with Phytoplasma infection. So keeping this in view, we have explored some strategy for identification of the Phytoplasma infection in the field at an early stage. So you are well known that we are all well known that volatile organic compounds released by the plants serves as information sources of infection, and these involve some plant defense chemicals and anti-herbivore communication chemicals, etc. So to explore the profile and the expression release strategies, release dynamics of volatile organic compounds in Phytoplasma infected plants, we have collected Phytoplasma infected plants, fully grown plants before flowering stage, and confirmed for Phytoplasma infection, and healthy plant which was confirmed for negative for Phytoplasma infection were taken, and also the scope for other infections were also eliminated, and static sampling was done through solid phase micro extraction, headspace GC vials, and the further analysis was done through predetermined protocol using GC TOF-MS. And you can see here the differentially expressed volatile profile from both healthy and infected plants. Many compounds are expressed in the same way, but there are some differential expression also. And out of the 68 compounds we have identified expressing in healthy and infected, 26 were differentially expressed. And I have given here the bar diagram of differentially expressed compounds. You can see here the orange indicates the infected plants and blue indicates the healthy plants. So compounds like methyl salicylate, linalool, isoborneol, bisabolin, philandrin, etc. were expressed only in infected plants, whereas some compounds like alpha terpenols, alpha pinene, limonin, etc. were expressed in both infected and healthy plants. And these compounds were further checked for their activities and their significance in the plant system, and we could identify most of them were sesquiterpenes and monoterpenes associated with plant defense signaling, and the upregulated, expressed released compounds like bisabolin, myrcene, philandrin, etc. were either attractant or repellent to the herbivores, or they had antimicrobial activities. You can see here also there are anti-aphid compounds and anti-herbivore defense compounds, as well as some compounds are plant defense. And here we have done analysis of different stages of the plants. The result I have not shown here, but from the little leaf early stage as well as during the phyllodes stage also. All throughout the stages we could find this compound linalool, which is acting as a leafhopper attractant, is expressed in high amount in Phytoplasma infected plants. So this may be a strategy of Phytoplasma to attract its vector for further spread of the Phytoplasma. And there are other compounds like aphid repellents were also aphid and whitefly repellents were also highly expressed in the infected plants. So this may be a strategy to keep the other sucking pests away, and only its vector is attracted by the plants. So this information can be explored for both the highly expressed compounds may be used as biomarkers for the identification in an early stage, and the other dynamics of the expression profiling may be explored to identify the mechanism of Phytoplasma infection in marigold plants to employ management strategies. So to conclude, it has been identified that volatile profile of marigold is affected during Phytoplasma infection, and the symptoms were symptom-associated expression profiling of volatile organic compounds were successfully done. Linalool, isoborneol, bisabolin, myrcene, philandrin, methyl salicylate, etc. were released in high amounts from infected plants and were very negligible or absent in healthy, while neophytidine, caryophyllene, camphene, benzaldehyde, benzene, acetaldehyde, etc. were absent in infected plants while found in healthy. And these identified compounds expressing only in infected plants has the potential for biomarkers for identification of Phytoplasma infection in an early stage from marigold through non-invasive diagnostics technology. And this can be applied for identification of mother plant stocks as well as for screening the nursery stocks produced from the mother stock for Phytoplasma infection. And understanding these changes in the volatile profile can be used to identify mechanisms of Phytoplasma infection to employ management strategies for Phytoplasma. And in future, I have done the static sampling of the volatile compounds. If we can do real-time dynamic sampling, we will get more accurate detection of volatile organic compounds. And from the identified biomarkers, if we can make some biosensor for identification of these biomarkers, then through ENOs or other miniature devices, we can employ the detection, on-field detection of Phytoplasma in an early stage. And diurnal variation also has some influence on the volatile organic compound release of during phytoplasma infection. So this can be explored, and in the field there will not be individual infections. So mixed infection scenarios has to be explored while we in future to see the expression patterns and host pathogen phytoplasma, host phytoplasma vector interactions study through this volatile organic compound give promising result for management strategies. I would like to thank Indian Council of Agriculture Research for giving me the infrastructure and Department of Science and Technology for providing me the funding, and my co-workers and co-authors for successful execution of the research work, and the organizers of IPWG, and especially Dr. Asunda and Dr. GP Rao for giving me the opportunity to present virtually. Thank you.
So thank you, Prabha, to attend us. And is there any question? Can I ask you, Prabha, for no? So can I ask audience for any question to Prabha? I'm not sure if Prabha is online still, but it looks
yes,
like
I'm on.
Yes, she is there. So if no question, thank you, Prabha, and I think we can go for a lunch. So
thank you.
Thank the audience, for all presenters for their interesting contribution, and have a good lunch.